Assalomu alaykum hammaga. Yana sizlarni ko'rib turganimdan xursandman. Va nihoyat biza 3-kunga yetib keldik. Good morning, everyone. It's wonderful to see you again. And we are today starting our third day of workshop. And I can see over the 2 days, we have shared wonderful presentations and thoughtful discussions, and it was really engaging. And I hope we continue with this energy. And today, you will be going through microscopy and cell biology uh, um, topics. So I'm happy to invite our today's first speaker, Michael Melzer. He's head of cell biology group at the Plant Institute of Plant Genetics and Crop Plant Research in Germany. And Michel, yeah. Stage is yours, please. And yeah, thanks so much.
So don't leave the stage, Bruno, because I really be happy to be here. And I think Bruno did really a great job. Everybody who knows him, he's very enthusiastic. And I think that's what we are as well. So it's a really, really nice country. And great hospitality, and a perfect organized sausage. You can see our shoes. Fair and red color. She wears the only red color because I sticker, and she will change that to black color maybe. Okay.
Thanks so much.
Okay, so we totally switched the topic. So, and the intention in my first topic is to show you how important, or I try to explain you that microscopy is quite important for a lot of fields in plant research. And nevertheless, you don't need always the real top fancy microscopes to do good research in microscopy. So, and I hope that I can explain you also that with a little bit basic things, you can do also good research. And maybe this is a little bit important for people here. If you're not really have all the fancy microscopes, but I think you need a little bit basic stuff to do really good work. So you can see that the location of your institute and our institute, there is a big distance. But I hope that the workshop will make it a little bit shorter and we come together. So that's the wish, and I have the feeling we are on a good way. So just a little bit about the IPK itself. So that's the institute. You see a quite big institute, 560 employees, 180 scientists, and at the moment, 32 research groups and 33 nations as well. So my department is the physiology and cell biology, and it's headed by Nico van Wireen and assisted by Ricardo Gil of the plant nutrition group. Then Hannah Schneider, and she's working mainly on roots. On the other hand, now a junior researcher, it's Robert Hoffy, also involved in transformation things. Then Yong Wang is also a new group, and he's working a lot on the topic. I will talk later in the research about vascular tissue. And finally, you know this guy as well, Jochen Kumlin. And our group is located also there in the center of the main building for the cell physiology. So I think I missed, so the world of imaging. So you come from the macro to the micro. Okay, micro is a little bit shorter than it has been shown here. And You can do it via satellite imaging, plant research, also aerial view from the helicopter. Unfortunately, at the IPK, we don't have a helicopter. So this is not available at the IPK. But what is available at the IPK? So there was a AgRover used with hyperspectral camera in the field. We have drones, of course, as usually in the meanwhile. We have the Phenosphere and LambdaTech devices to do automatic plant phenotyping. And also, on the other hand, in the group of estimate allocation and NMR, we have the possibility to do non-invasive structure analysis. For example, here you see barley. And if you check the barley kernel a little bit more in detail, you can get information about the different tissue, embryo, the cavity, and the endosperm, and finally the pericarp. So microscopy at the plant research and at the IPK Gardaslim. So the structural cell biology group has conventional light microscopy. Then we're doing histology, fluorescence microscopy, and also digital microscopy. Then on the other hand, confocal microscopy. So we have laser scanning microscopy available and also spinning disk. I come to this a little bit later, what the difference is. Of course, scanning electron microscopy, but there you can do just surface analysis. You can do also immunogold labeling, and you can 3D reconstructions in the meanwhile. On the other hand, transmission electron microscopy with immunogold labeling, ultrastructure analysis, and as well 3D electron tomography. So additional, in another group, we have the so-called light sheet microscopy, which makes it possible to analyze bigger structures. But a little bit the disadvantage is that you have to have a sample which is more or less transparent. And then you can imagine, okay, for a leaf sample, it would be difficult because it's not transparent. But for roots, it's working perfectly. Otherwise, you have to do a clearing so that your sample is transparent, more or less. On the other hand, super resolution microscopy in Andreas Huben's group, and you can see some images. You can really see details of the chromosomes and the labeling of the distinct areas. So, but there are many, many other microscope systems, and this is just a little collection of it. So, for example, two-photon laser microscopy, or squid microscopy, ultrasonic microscopy, where you see on the right-hand side an example. And in an electron microscopy field, there are also a lot of more instruments. And of course, we don't have all of them at the IPK. But now I will show you what we have at the, later what we have at the IPK. So just the basic thing, most of the people have used the microscope during their studies. You have a light source, you have a lens, your sample, and you see something through the eyepiece or in the meanwhile in the digital camera. So the confocal works a little bit different. You have, instead of light, you have a laser beam. This is exciting your sample. The fluorophores, you will get an emission. This is going back into distinct detectors. And it's also a possibility to go to a special detector. And this one is doing spectral imaging. So it's a spectral separation of different signals. And here, on the other hand, you have a confocal microscope, which is the spinning disk. Here, the difference is you have one laser beam. You have a rotating disk with a lot of holes, up to 1,000. So the laser beam is split into more or less 1,000 single small beams, and then you excite your sample at the same time. So this is shown here, confocal, line by line, the scanning, you will build up your image, and the difference with the spinning disk is you will get it immediately. There is a little bit a difference in the resolution, but the advantage for the spinning disk, you can image very fast events in the cell quite nicely. So there are two natural enemies in fluorescence microscopy. The first one I didn't mention is a bad user. But so one enemy is photo bleaching. If you have too much power on the laser, or it takes too long time, then your fluorophore is just bleaching out. You get no signal anymore. So this means that you have to do it as faithfully as possible. Just use the laser power what is needed and do it as quickly as possible. On the other hand, autofluorescence, which is shown here in red, in that case, it's really helpful, get a nice image. But very often you have overlapping signals with the autofluorescence, and this makes it a little bit difficult to discriminate the signal what you would like to have and about autofluorescence. For a scanning electron microscopy, you have, instead of a laser or light, you have electrons, and the electrons are hitting your sample on the stage. So you have two events. You excite electrons, secondary electrons, and you will detect these secondary electrons. And on the other hand, some electrons are backscattered from the sample, and you will detect it with a so-called backscatter detector. So transmission electron microscopy is, as the name says, it's transmitting your sample, and the electrons which going through the samples will be then collected on a screen, on a fluorescent screen in older days, and in the meanwhile only by digital cameras. So you have a lot of nice images what you can get with the different systems. But there is one question. So, but what do you really see? Is it the truth, or is it a nice image, but has nothing to do with the real event in the cell? And here I have the only part of Russian, the only Russian part in my talk, just to explain you this important question. That's it for the Russian part, sorry. So the key and really the bottleneck for microscopy is sample preparation. And this is really, really a dogma. So first I will talk a little bit about use of fresh samples. You can use it with the digital microscopy, then also with the confocal and with a light microscope. Here you just see some examples. On the right-hand side, you can really nicely see also here the pollen here at the anther and so on. So this is how it looks like. You don't have eyepieces, so it's just a camera here in the center, here, here. And the advantage for this type of microscope is, okay, it stops. Okay. These are the examples, for example, because it's important to get a general overview of the sample, that whenever you're using a stereo microscope, you know that you face the problem that you can get only one layer in the focus and the rest not. Here, you just have a click and the whole sample is within a second in the focus. And you see also the resolution is quite nice. You see the trichome of Arabidopsis. So in that case, you don't need to have to do a big preparation to go for the scanning electron microscope if this resolution is enough for you. Then you have also an on of body, the leaf surface of violet, which looks quite nicely, and hibiscus flower also with pollen, and the leaf surface of liverworth. So then it can be used for some measurements. Here, for example, we were interested in the thickness of the internodes of barley samples, or you can check the architecture of roots. Here on the right-hand side, you have a root mutant with short root hairs. And on the other hand, you can use also sections from histology just to figure out how big is the intracellular space in comparison maybe to the mesophyll and the mesophyll cells. This is quite important to get this information about photosynthetic rate and so on and so on. It is quite easy to be done in the microscope. So there is another techniques. If you would like to go a little bit more in detail, you can use fresh samples and prepare sections. So therefore, you have just simple tools. You see some slides, a razor blade, forceps, and in sometimes agar. If you have a very stiff sample, you can use it directly, but otherwise you have to use agar. For example, just to cut off a little bit of the internode or the nodes, bring it in agar, then you can cut the agar block and fix it with a super glue on your sample holder, then go to the vibrotome, and you can prepare sections that depends on the sample in a size about 50 to 300 microns. So what you can do then, you can do some labeling here, in that case, fluoroglycinol, or on the upper panel, it's uncontrolled control, and on the upper panel, you can see then after a specific staining for lignin. So unfortunately, the presentation is a little bit weird, what I realized in the meanwhile, because this should become before. Anyway, you can see how it goes. You have the sections, then you transfer the sections into a container, and then you add your staining solution, and you would get your stained samples, and you just go to the microscope and check it out. And then there was the other image as before. Let's go to fluorescence microscopy imaging, live cell imaging. So we have three systems in the meanwhile, the Zeiss LSM 980, and with the so-called Aeroscan on the left-hand side. And this is a device which makes it possible to analyze your sample in a much more sensitive way than before. We have the LSM 780 and the Andor resolution spinning disk system. So as Jochen already explained about haploid technology, so he showed some nice movies, but this was the end of the project where we established everything. I will just show you now how we established that system to get these images. And in the first run, we tried to keep the cells alive for a long time. And the video what you see now was taken over 30 days. One stack each 15 minutes. And in totally, we applied more than 50,000 images. And hopefully, okay, now you can see the movie. And this is a movie which really covers 30 days. And okay. I really apologize for the quality of the movie, but this is maybe due to the system. Okay, so the challenges for the project was really the constant provision of effective pollen density. Then you have to have a reliable cell immobilization so that they are not traveling around, which is quite important. And it's important to have the right concentration of the culture medium and the excess of the feeder molecules, and of course a sufficient gas exchange. And then it's the monitoring at high temporal and structural resolution, of course, is important to get those images. And this is how Dia Dagmar did it, the PhD student at the time in the group. And he just made a self-made chamber. And this is how it looks like. So this is just a counter cell counting chamber slide, and he modified it to the needs. And this is how it looks like. And so he was really able to create and optimize the experimental setup. And I just show you why it is important. You see here the collection of the viable cells. Then you have to transfer them and into the chamber, then do the live cell imaging. And then finally, you get these images that Jochen has already shown. And I just want to show you a nice example. That's not always, the result is not always what you expect. And here, just watch the nucleus in the bright green. What happens? So it tries, to divide, but somehow he's not really successful. And finally, he gave up and simply exploded. So this is also another example where we're able to show by our confocal microscopy that the parental DNA was just thrown out of a crossing between wheat and pearl millet, and this was done with the confocal, and this was the starting point of virtual reality because we did it with the Fraunhofer Institute. Then you go into a room and you really walk in between the different cells and to have a look into the nuclei. This was quite exciting, but it took a long time to get all these samples together. So the fixation of plant tissue for histology and TEM, this is really, really a very important part. So because that's a fresh sample. And finally, after fixation, you would like to have the sample in the same way. You don't want to have something like that. But this happens very often. And the problem is, if it's not so severe, you don't realize it, especially if you maybe compare the wild type with a mutant, because you expect, okay, the mutant looks a little bit strange, but maybe it's the fixation also. So be very careful with that. So the goal is really to preserve the structure as faithfully as possible compared to the living state. This is very, very important. So just an example, for example, here a barley plant, we cut off some small leaf pieces, and this is used for fixation in aldehydes, usually used for 2% formaldehyde and 2% glutaraldehyde for ultrastructure analysis. And if you would like to do immunolocalization, you use less glutaraldehyde because it protects your antigenic sites in a better way, and it's better available for the antibodies. What we are using also is a microwave system. You can do the same steps very fast, and it's interesting because all the molecules are in moving, so they vibrate, and this means you have an increased diffusion rate for the fixative into the tissue. Then increased temperature is also more efficient for the fixation, but it's really mild temperature. It's not like a normal microwave. Otherwise, you would cook your sample. And this is also awful, finally. So, and what was really surprising for me as well is that you have an increased structure preservation. So, and the most important thing is also that you have really time savings, because usually I would say that the conventional fixation, just the fixation takes about maybe 1 day. And here you can do it in 2 minutes. Yes, in 2 hours, sorry. Just an example, this is typical vesicle formation that you see after chemical fixation. And this is how it looks like after microwave assisted fixation. It's a little bit similar for all the other structures. So what happens after the fixation is that you do a resin embedding. You have different containers. For ultrastructure, for example, we are using very often the spur resin, which is shown here. And what you can see is also that the leaf sample turns a little bit into black. And this is because we are using, after the aldehyde fixation, secondary fixation with osmium tetroxide, because osmium tetroxide is a heavy metal, and this is increasing the contrast in the electron microscope. So if you do histology, it's not necessary. But for pure ultrastructure research, osmium tetroxide is important. But if you would like to do immunolocalization later on for proteins, then you should not use osmium tetroxide, because osmium tetroxide is disturbing your antigenic sites. You don't get a signal. Ella White is used for both, for immunolocalization and also ultrastructure. And here on the right-hand side, the HM20, you see also the leaf tissue, but it's more transparent. And this is exactly the difference. So then the next important step is to cut it into small slices as you cut the bread, but of course much thinner. So for histology, you can see it's about 2 to 5 microns, and you do it with a microtome, and if you can afford it, with a diamond knife. And you get semi-thin sections about 1 to 3 microns. You will transfer it on slides with a little bit water, you dry it, then you can do a staining and then mount it, and then finally go to the microscope and check, for example, here the root primordia of Petunia. On the other hand, just some applications. This is from a maize root with differential interference contrast. It looks really like art. This is a xylem tissue, or what we have tried. And because there was a demand to show the gas staining really precisely in the cell, because if you do, everybody who's working with gas staining, knows there is diffusion into other cells, and sometimes it's difficult to distinct the staining in the different tissue. And we tried it with histology just very quickly with a microwave, and finally with section, and it looks really nice. And then, for example, leaf beach, just a cross section. And here we were interested also in the density of the vein tissue. We did some analysis here, and it depends always how you cut your sample. Again, back to the pollen embryogenesis. What we have done at the beginning also is serial sectioning on a histological level. And then you get also a nice 3D reconstruction. There is one cell here on the right-hand side, now moving to the left, where you can already see the chromosomes. So this is done really by light microscopy, serial sectioning of histological samples. So then digital microscopy we are using very often here in just one example, chloroplast biogenesis in barley. You have your sample, you just put it under the microscope, and then you can check out the surface and the pattern of the leaf. And here it was a mutant with problems in chloroplast biogenesis. I come to this point a little bit later again. So this was with Niels Stein. So this was and we would like to characterize this HVAST mutant, which is a problem in chloroplast biogenesis. So sectioning for TEM, it's a little bit the same, but you have much. Here you can see it. You cut the samples into slices which are about 50 to 70 nanometer, very thin, very sensitive. Of course, you have to transfer then the samples onto the grids, the copper grids, which are at about the size of 3 millimeter. And this is a very sensitive procedure. And then nevertheless, if you check it through the eyepieces, you can even see the leaf structure on this very, very thin sections. And then you transfer it to the grid. And what you can see also, you have to be very careful. You have to work very precisely, because otherwise you have holes in your sample. These are just some examples. On the left-hand side, Arabidopsis nucleus and a metaphase, then a maize chloroplast in the center, and to the right, plasmodesmata of a tobacco plant. Back to the cooperation with Niels Stein. Here we have used different type of microscope. On the upper panel, the light microscopy, then electron microscopy with a detail, just to check out the chloroplast in detail of the different mutants. And this is a yellowish one where you can still see some granostacks, but totally different shape and very less amount. And on the right-hand side, this is the albino mutant where you see that the chloroplast is not able to develop any membranes. And what you can do as well, and we have done it here, just checking out the shape, the chloroplast area, for example, we measured the length, the height, and the number of granostacks, and then in detail, the number of granostacks, and then number of the Grana-Stack thylakoids, and we did also measuring how then calculations and so on and so on. Another very important method for us is immunolocalization. So this, you have your primary antibody. In most of the cases, it's IgG. And what is recognized by the secondary antibody is the upper part, the Fc fragment. And this is how it goes. You have your antigen, for example, in the membrane. Then your primary antibody will bound to it if it's the right antigenic site. And in the second hand, you have the secondary antibody, which is recognizing the primary antibody. And you have a gold particle like a rucksack. And this is the gold particle is the only thing you can see in the microscope, finally. Because otherwise, all the proteins still look more or less, they have the same pattern in the electron microscope. And this is how it looks like. An example for Rubisco labeling, of course, it's highly abundant. Or here you have copper zinc salt dismutase in peroxisomes of a pine needle. And another technique with the electron microscope is to get also a three-dimensional information. But with this 3D electron tomography, we are limited to 200 to 300 microns. So that's not so much. If you see the dimension of a chloroplast, it's about 10 microns. You can get only a part, but it depends on what you would like to show. If you would like to show the membranes a little bit in detail, it's fine. If you would like to show the whole chloroplast, not possible. So it's just, I just have to go back. So what will be done is like tomography, also in medicine, you just turn around, you tilt your sample, take an image, tilt again, take an image, and so on. And then we have a 3D reconstruction at the end. And I will show you one example where you can see that it's sometimes important to check your sample again. So what we have here is HK9 protein that is abundant in the mitochondria. It was fused with GFP to check out at different development stages how it is. And so we checked it in a microscope, and you can see the small green dots. The size is okay for the mitochondria, so the cooperation partner was happy. And then I said, okay, but we have also bigger ones. And the bigger ones are too small for a nucleus, because nucleus has a tendency to catch the GFP very often. So we thought, okay, we have to check it a little bit more in detail, because we were not sure what it is. We were thinking about it. Okay, and then this is the size of a nucleus. It's too big, so it's not a nucleus. Mitochondria, too small, so not also mitochondria. But then we detected some really strange structures I've never seen before. So then it looks like, if you should check from the size, then you maybe think, oh, these are kind of mitochondria. But what happens? It looks like cancer, or they are stacked together, so something happens. And the first thing was, okay, we have to verify that these are mitochondria. So we did immunolocalization with glycine decarboxylase, which is abundant in mitochondria, and we get nice signals. So we thought, okay, the confirmation, these are mitochondria. And now it was a question, are they stacked together, or is it kind of a cancer? And if you check a little bit carefully, this is just the first movie, it has the tendency that now we are going through layer by layer, that they are not fused. And you can see that nicely in this movie, if you check a little bit carefully. So we saw that these are really stacked mitochondria, but really monster mitochondria. And the good thing was the group with the cooperation partner, they checked the trends, the vectors and so on. And so they realized that the construct was faulty, and they did it new, and then they solved the problem. So there is another technique, and this will come in next March. We will get a new electron microscope. And this is a so-called scanning electron microscopy STEM. So you're doing a little bit the same, tilting your sample, a different image from different angles. And the advantage is that here you have your copper grid and you have parallel electron beams. So this gives you a then better resolution than in a transmission electron microscope. And the big advantage is also you can use samples up to 1 micron. So this is much more, and this is really helpful. And that's just how it shows. We have to use a bright field detector, and there is a special detector at high angle, so-called high angular dark field detector, and this is collecting electrons that are also scattering at high angles. So we get much more information than with a normal TEM. So what is very important that the sample is always at all tilting stage in the focus. And this is a little bit different to the 3D electron microscopy before. So just on the left-hand side, a very easy example from one of the demos, which shows you a little bit part of a chloroplast. We did not do a segmentation here. On the right-hand side, I have a friend at the university in Regensburg, and he's a real expert in STEM tomography, and he gave me the example here of a human retinal pigment, epithelium 1. And what you can see here, they are going through, and then they will demonstrate what you can do out of it with segmentation stuff. And this is really at high resolution. This is really a big advantage and a very important point for the future. And maybe in 2 or 3 years, I can give you some examples from plant research. Okay, a lot of nice images out of electron microscopy. And also here is the question, what is the best sample preparation method? You can do the chemical fixation, the microwave fixation, but it points out that high pressure freezing is the method that you have to use. It's the really cutting edge method in sample preparation. So you can see a big machine. It's really a big machine. It makes really noise because it's working with 2000 bar. So this means you have a very small sample and you really push the liquid nitrogen with 2000 bar onto your little sample. This is how it goes. So the first, and we did it also for pollen embryogenesis in that earlier project. And unfortunately, we realized that it was horrible at the beginning. You see, this should be the pollen, and they look totally horrible. And at that time, yeah, it was really disaster, and we thought it was very important for the project. So how to solve the problem? And then I remembered we had a project with cyanobacteria, and there it was working perfectly. You see it really, really nicely, the membranes, so everything like drawn with a pencil. And so I told dear Dagmar at that time, we have a good one and we have a bad one, try to mix it. And what we did, we filled, the way this is a nitrocellulose tube, just to have your samples in a kind of a package. And then we filled it with cyanobacteria and did the same thing as before. So this is how it looks like. You load it with capillary forces and cut it into small sausages or small packages that you don't lose your sample between all the following procedures. This is how it looks like. You have the aluminum platelet, which is just a diameter of 3 millimeter, and the thickness is 2.2 millimeter. And we should not use more than 2 because we have platelets with 0.3 and 0.3. You can combine it and then you would have 0.6. But physically, it was shown that it's not working with this procedure because otherwise you will get ice crystal formation. Ice crystals will destroy your sample. And therefore, we have to stick to 0.2 millimeters. And you can imagine for a lot of samples, it's not so much. But here you can see the result then after high pressure freezing. So it's really, really nice samples. And if we And now to show you also the difference between high pressure freezing and a real good microwave sample on the left-hand side, but you will see if you check in the vacuoles, you will see that there is a difference. Point out here. You have, after high pressure freezing, you can see that there are a lot of invaginations into the vacuole, and they simply disappeared in the chemical fixed sample, although it's a good sample. And of course, there is a difference in some organelles. You can see much better the nuclear membrane with the pores and so on and so on. So we were quite happy about that. Another thing is also high pressure freezing is really helpful if you have immunogut labeling. Sometimes you do immunogut labeling, you get your antibody, you do it, and then you realize, okay, no signals. And the question is why? And sometimes it's just the preservation of the antigenic sites. If you do it with high pressure freezing, so first I will show you here just an explanation. You have bundle sheath and mesophyll cells, and the localization of this glycine decarboxylase is different in mesophyll and in bundle sheath cells, and we would like to show it. And here we have the labeling, and there is nothing. So then we did also for the high pressure freezing, nothing. But this should be the case that for the mesophyll cells, there's no labeling. But we should expect in the bundle sheath labeling. And this was a difference. And this is a running project with CEPLAS in Düsseldorf, with these two guys, Jose Valderrama and Patrick Lederer. And the main task is immunolocalization of different proteins and also morphological and structural analysis about mitochondria, chloroplasts, and so on and so on. So just a few other examples. Chemical fixation looks nice, I think. That's after high pressure freezing. You see the plasma membrane quite clear. Mitochondria are more round shaped. And also the cell wall, you see the texture much better. And for example, here also the tapetum cell and the microspore, you see the difference on the right-hand side. Sorry, and here maybe a pollen as well, nicely preserved, dictyosomes, mitochondria, and so on and so on. So we were really happy about that. And then try to get the perfect one, the perfect sample. So here you see also there is a difference. We check under the light microscope, and the lower one was not really good working. So this happens also, but the other one looks quite nice. And then in Moricandia arvensis, this is the plant we are working on, this C334 metabolism, looks quite well. So we checked it in detail, and it looks really good under light microscope. But sometimes it looks good under light microscope, but if you go to the TM, it's totally frustration. But here it's the opposite. Everything looks really beautiful. Here you see also the nuclei, and so everything looks really fine. And also the chloroplasts, you see the membranes quite nicely. And here also some microtubules, the plasma membrane, dictyosomes. And the question was, did we find the perfect sample? No, because this can happen also at high pressure freezing, that they disrupt. I would say we're not 100% sure if it's really a disruption due to high pressure freezing, but this can happen at high pressure freezing. Okay, so just switch a little bit to electron microscopy, to scanning electron microscopy. You do the sample preparation as well. You have a sample container, then you have to dry your sample in the critical point dryer. Then you have a dry sample. You have to sputter it with gold, and then it looks like that. And then you go to the SEM, and you will get nice samples here. Barley spike. After development, you can color it if you want, because it looks a little bit more fancy. Then here are Arabidopsis trichome, for example, xylem barley. Then leaf surface with stomata, we use it very often for counting the density of the stomata. And a pistil of Arabidopsis with some pollen around. Then, for example, a wheat chromosome after immunogoat labeling, and typical Arabidopsis seed. And if you have time and mood, and you can color it so that it looks a little bit more art style. But this is just playing around with no scientific relevance. So on the other hand, there is a possibility to do also 3D imaging. So what you're doing is also cutting. You have a very tiny microtome in your microscope. And what you are doing is you're just cutting off, you're imaging the surface of your sample. Then you cut it off and you're imaging again the surface of your sample. And here with that technique, you can get much bigger samples for 3D reconstruction. So it's not a problem to get a chloroplast or even a whole cell into the images. So this is how it looks like then finally, for example, here with chloroplast on the right-hand side. And that's how it goes. Then you can go into the different layers of your sample, and you can do some segmentation later on of the membranes and so on and so on. There is also another technique which we don't have it at the RPK, and this is, I would say, the best to get fine structures, because this is a so-called focused ion beam scanning electron microscope. So you have your sample and you have a fine ion beam and you mill away layer by layer and then take also the image from the block face. And here on the left-hand side, we have our sample that was tried. High pressure freezing, of course, is here also the best. And then on the right-hand side, you see how it looks like. You have a part of the nucleus here imaged by focused ion beam. And this is really, really at higher resolution. And then finally, you can do also the 3D reconstructions. Last but not least, there is X-ray microscopy. We do it in cooperation with the Fraunhofer Institute in Würzburg. This is sometimes very helpful because you can do it quite quickly, that you get 3D information, of course, not at the same resolution level. And you see your sample here. This is part of I will come later to that part again in my research talk. This is about the Caryopsis, because we were looking for a so-called abscission zone. On the left-hand side, you can see what we have done with histology work, and on the right-hand side with X-ray microscopy. And oops, hopefully, let me see. Okay, I have to do it in an old-fashioned way. Okay, so that's what you can see here. This is done with histology work. So, and then we just checked out the abscission zone, and it's very loose here. So this means that this seed is going off very easy because it's not well connected to the tissue. And on the right-hand side, X-ray microscopy, which takes maybe 2 days, and the other one takes 3 weeks. And that's a big difference. From the resolution, I would say there's not really a big difference, and the result was exactly the same. Okay. So, whoops. Hopefully I was able to show you the microscopy is really an important link in plant research, because it's always needed in a lot of purposes. So this is my take-home messages. And nevertheless, it's important to do this work to have a really nice group. And we now, 3 of us are working together 26 years now. And you see them with the arrow, and slightly changed after a long time. And this is more or less the actual team. Kirsten Hoffi and Marion Bennecke, our technicians, Claudia Riemey and Sibylle Freist as well from time to time. Bartjor, where's Bartjor? He was helping us tremendously in one project. I will come to that later. And then Twan Rutten, of course, who was doing all the confocal work and scanning work, and then Stefan Ortleb from another group at the IPK, and Felix Rose, who are supporting us really greatly with the 3D reconstructions. And the last slide is this, and this is not a fake video. And hopefully that not everybody was sleeping during the talk. I hope you're a little bit excited, and it's not always like that. I hope that it is starting. Okay. So the first one, second one, third one. And this is a real video. This was taken by my girlfriend on a visit to South Africa. Okay, thank you very much for your attention.
Yeah, very insightful and impressive image. Thanks so much for the clear and informative overview about the microscope and techniques. And so now the presentation open for discussion. Anyone has a question?
Thank you. That was a nice tour de force of microscopy, I guess. Are you doing any super resolution microscopy on the fluorescence?
Sorry?
Are you guys doing any super resolution microscopy?
Not ourselves, but the group in Andreas Ruben's lab. So it's the problem with the super resolution microscopy is if you would like to do it in light cell imaging, that's a limitation very often. And we tried. I've shown you this example with the hexokinase 9, and we had some similar with a movement protein in virus infected cells, and there we were clearly shown with immunocode labeling that they are stick to the plasmodesmata. And we tried that also with super resolution microscopy. We didn't see anything.
Were you using 3D sim, or like what were you using?
Yes.
Okay.
سوال بارمه؟ اگر kimdir savol bo'lsa bemalol agar uni tarjima qilamiz. If not the case, thanks so much, Michelle. One more. So we move to our next speaker, Professor Stefan Persson from University of Denmark, Copenhagen, Denmark. So his research focus on cell biology and cell wall and its organelles and components. And yeah, stage is yours.
Thank you. Yeah, thanks, of course, to the organizers for inviting me. Arno, thanks so much for having us here. It's been a very interesting conference for me. I haven't understood fully most of the other talks, some very well, others maybe not so much. So our lab is mainly focused on, of course, cell wall synthesis, but we're taking a very much of a cell biology and molecular biology based approach to this. So in the first talk, I'm going to talk more about cell biology, so a lot of microscopy. So thank you to Mikael for introducing all of these microscopy techniques, because it makes my talk a little bit easier, I guess, as well. Really, what we're going to talk about in this talk are these two things, basically. So we're going to, I'm going to give you examples for cell wall synthesis in growing seedlings. So basically, like how seedlings direct their growth. And in the second part, we're going to talk a little bit about wood formation. Both of these systems are really like cell walls are central to the development of those. So cell walls, you probably think about this, right? When you hear cell walls. So it's kind of like the walls that are surrounding us in this conference room as well. You can think about the walls as the cell walls, right? And we have the cell in the middle, right? And so with this aspect, it might be not so difficult to also put this in a pathogenic context, as we heard previous talks. Of course, the cell walls are there to defend the cells against, for example, biotic and abiotic stresses. Of course, the cell in this conference room would need to grow at some point as well, right? I mean, we know that cells are expanding. And if the walls would look like that, of course, this would be a disaster, right? So the walls would move, this roof will fall in, and we'll probably all perish, right? So cell walls needs to be able to expand. They need to be dynamic. And this may be illustrated over here as well. So there you see a cell, basically, and the cell walls are dynamically put down around the cell, right? And you can perhaps envision this as the cell walls here mainly being laid down transversely around the cells. And of course, now when the cell expands, it has to expand in the longitudinal direction because the walls are preventing expansion in the other direction. So this is also giving shape to the cells and of course also to the plant. Right? And actually, my previous career was actually in construction engineering. It's far way back. It's probably telling you how old I actually am, right? But I think there's a nice connection between actually the walls that you see to the left-hand side and the cell walls that we're currently trying to investigate. So cell walls are not only important for plants, and they're of course also quite important for us. So for example, a lot of you are working on cotton, and cell walls, I mean, cotton is virtually just cellulose, right? And that's a cell wall component. It's also important for the woods that we are having in, well, basically what I'm standing right now, yeah. And of course also for fuels. We had this like second generation biofuel hype for a while, but cellulose was going to be degraded into ethanol and then fuel cars and so forth. It's actually also important for food and feed. For food, we heard before, like the fibers for the beta glucans, for example, in terms of actually your digestive tract. But of course, most of our cows, pigs, and so forth are actually eating plants and therefore cell wall material. And finally, it's actually our major climate change mitigating resource because most of the carbon dioxide that is fixed through photosynthesis in plants are then being deposited into cell walls. So if we can just make the carbon dioxide stay in the walls, then of course that would be very beneficial. And so maybe the last aspect of why I think it's important to study cell walls is that these polymers make up the most abundant material on Earth. So maybe we can go into like how these cell wall polymers are made. So most of them are actually made inside of the cell. Let's see if this is working. Inside of the Golgi in here. So here you would have substrates, so nucleotide sugars, being transported into the Golgi lumen. And then basically these other enzymes in here, transferases, are stitching these sugar molecules together to form sugar polymers. And those sugar polymers are then transported from the Golgi to the cell surface where they're kind of baked into this growing cell wall. There are, of course, also exceptions to this rule. Actually, the thing that we're mainly studying in my lab is actually occurring directly at the cell surface. So you can see here that you have these cellulose synthase proteins that are making cellulose directly at the surface. So plant cells are investing quite a lot of energy into making these cell walls. So it's estimated that 5 to 10% of the typical proteome in a living cell is dedicated to making these, changing them, or breaking them down. And so if we now skip out to the cell wall, this is typically what you would look at, right? So you have this kind of almost like a jungle of sugar polymers that are now dynamically interacting with each other. And here again, you can see these cellulose synthases that are making cellulose fibers. So in our lab, or in my lab, we're interested in a range of different questions that are related to cell wall structures. So we're interested in what the thousands of proteins that are associated with cell wall production actually do. We're interested in how these proteins and the corresponding genes are regulated. We're also interested in how they engage with each other and where this is happening. We're interested in how cell walls can communicate to the cell. Now, it has to basically tell the cell to make more or less cell wall polymers. And we're also interested in how cell walls really look like and how this relates to how plants grow. And finally, we try to put this in context of development, environment, and also evolution. And today's talk, we're mainly going to focus on this, so basically the development of different organs and so forth, and how cell walls are contributing to that. So to understand that, we first need to look a little bit more at how cellulose look like. So it's a quite simple polymer. Actually, it's just like glucose units that are stitched together in this like beta-1,4 linkages. And then they actually align with each other to form hydrogen bonds, and it makes these like very strong microfibrils or macrofibrils. And you can kind of see this as biological steel wire, because they actually have a tensile strength that is equal to steel, more or less. So you can now imagine if you wrap these cellulose fibers around the cell, of course, that is going to provide basically the stiffness or the strength of the cell wall, and therefore also like regulating the morphology of cells. So if you have this stiff cellulose microfibril coming out up here, and this cellulose microfibril is now baked into the wall. So basically it's immobilized, it cannot move anymore. What the heck happens when you make more cellulose here, right? I mean, if this guy, or like the cellulose microfibril, cannot move, what do you think will happen when you're making more cellulose here? Any bids from the audience? Do you think it will just be like that these glucan chains will like extend up like that and then going down, or like what do you think will happen? So it's probably most likely that the whole complex will move in the plasma membrane forward, right, while it's making this. And actually, through some of the tools that Michael was showing in the previous talk, we can actually visualize this directly in a plant cell. So this was done actually by the PhD student next to the lab, me in the lab in Carnegie. So I overlapped actually with Sue at Carnegie at the time. But he was making this construct. So basically, he fused one of the CESA genes, so cellulose synthase gene, with the fluorescent protein, and then he put that into Arabidopsis. And here you're looking at the basically growing, well, as a hypocotyl, right? And you looking at the single cell from that hypocotyl. And I hope you can appreciate that you can now follow the cellulose synthase complex in a living seedling while making cellulose, right? And this is pretty neat, I think. And we can, of course, now begin to understand a little bit more about how this process works, right? So, for example, you can calculate the speed, right? So it's moving with 150 to 500 nanometers per minute. And you can translate that to amount of glucose units that are incorporated per time unit into these cellulose microfibrils. Another interesting observation was some of you probably noticed that most of these dots are moving in this direction, right? Not in this direction. So there has to be something inside of the cell or in the cell wall that is steering where these cellulose fibers are being deposited, right? And so old electron microscopy imaging has shown, or like had shown, that cellulose microfibril that you see here in orange are typically laid down in parallel to internal structures that are called microtubules. So they are a major component of the cytoskeleton. And actually what you can do now is just cross our fluorescent line with the CESAs, as you see down here, with a microtubule marker. And I hope you can appreciate that these cellulose synthases are moving on top of these microtubules when they're producing cellulose, right? So this was an elegant way of showing basically the thing that had been shown with electron microscopy imaging before. So in a way, you can now visualize this process in your mind, hopefully, like it's this like kind of old steam engine train, where the train is basically the cellulose synthesis. The smoke could be like cellulose, right? And then you have these rails that are working as the microtubules. So basically, if you change these rails, you will presumably change also the direction of how cellulose is being produced. All right, so basically using microscopy, we've actually learned a lot about like how these cellulose synthases are regulated during this process. Right? So by basically like using a trick where you bleach a certain region of the cell and look at how fast cellulose synthases are repopulating the area. We can calculate, for example, how long the cellulose synthases are active with the cell surface. We can calculate how long the cellulose fibers are. And finally, we can count, of course, the number of sesase per cell. And so in a cell, we typically have about 2,000 active sesase in these growing cells that are basically making up these seedlings in this case, right? Okay, so I hope I've shown you here that we can really measure and evaluate cellulose synthesis in real time in a living plant, right? Okay, so now we're moving from these like primary cell walls that are basically supporting the direction of seedling growth to secondary cell walls, so basically wood-producing cells. And so there's some differences between these primary cell walls and secondary cell walls. And so primary walls that are mainly made up of these like sugar-based polymers, right? They surround each and every plant cell, and they're very dynamic. Secondary cell walls, on the other hand, they also contain these sugar-based polymers, but also something called phenolics or lignin. And they typically are made around or in woody tissues, and they're really stiff and strong. They're kind of the concrete of cell walls. And so you can appreciate that perhaps here, if you look inside of a stem of Arabidopsis in this case, you can here see we have stained just these secondary cell wall producing cells. So you have the xylem that transports water, and then you have these fiber cells that give stature to plants. And of course, these secondary cell walls were necessary for colonization of land by plants, right? Because they now allow water to be transported to the aerial parts of plants. But they're also key, of course, to biomass of the plant. So they're really important for a lot of different aspects for industry as well. Right. So just to give you an example here, actually, if you now look at water, it's transported through, in this case, a tree, right, up to the canopy. Actually, most of the water is evaporated. It's about 95% of the water that is transported up to the canopy is actually like evaporated out through stomata, right? But then you have this water that is now conducted through this system, and you can probably appreciate that as just cells that are aligned with each other and are dead cells actually that then conduct this water up to the aerial parts of the plant. So this, of course, needs to be quite strong, because you can imagine that this is almost like a straw in your soda, right? You suck on that straw, and you need to have a sturdy straw to actually support that, right? So if you don't have that, actually my postdoc, he was from East Germany, and he compared these straws that they got when they were drinking Cuba Cola, I think in East Germany at the time. They were not very sturdy. So when you sucked too hard, actually this straw is just like buckled in, right? So they basically, this pressure, inverse pressure that you're generating is basically collapsed the straw. So you wouldn't get like any Cuba Cola in that case in your mouth, right? And this is actually the case as well. So if you're mutating some essential cell wall components, that are associated with secondary wall synthesis, you can here see that you're getting these collapsed cells as well, right? And this is due to this like negative pressure that is generated. So to support this, right, both to give the strength, but also to transport water, you need to have like certain structures of these secondary cell walls. And you can see that they have different structures. You have this like, let's get banded or spiraling patterns, or also pitted, or basically like holes in the secondary cell walls. And so in our case, we were really interested in trying to visualize this process in real time. So basically visualizing wood formation in real time. And this is quite challenging because most of these wood forming tissues or xylem cells or xylem vessels, they're buried inside plant organs, right? So you have multiple cell files that you need to penetrate to basically get the imaging going. So in our case, we actually took a shortcut to this process. Basically what we did was to tell the plant to make secondary walls or this woody cell walls in all cells. And we did this by introducing this construct here. So basically this is VND7. It's a master regulator for secondary cell wall synthesis. So basically if you turn on that transcription factor, all of the cells will turn into wood in a sense, right? And so how this is working is that typically you express this VND7 here, right? But under normal conditions, so without this inducing factor, it's tethered to heat shock protein in the cytosol. And so then it cannot go into the nucleus and it cannot activate this process. Right? But of course, if you add this inducing factor, we can now like basically get this transcription factor into the nucleus. And therefore, you can now actually turn on secondary cell walls in all cells in a seedling. And just to zoom in, you can hopefully appreciate here that you have these bands that are now surrounding all of these cells in a seedling. And so basically now we have to go back to the first part of this talk, because I guess now we're actually moving away from making just these primary walls that are kind of evenly distributed across the cell to these banded patterns, right? And if we now remember the first part, then the cell wall, I mean, in this case, cellulose, they're basically guided by these rails, right? So our prediction here would be that the microtubule organization, or these rails, would change from this more random organization, or like actually like evenly distributed organization, to these bands. So what we did was just to introduce a microtubule marker in this VND7 line, and I hope you can appreciate here that over time after this induction, you can see this evenly distributed microtubule array that is now changing to this banded pattern, right? And now we can, of course, follow this in real time in a cell. So our movie is actually around 10 hours long, but we've cut it down here in interest of the coffee break. But you can hopefully appreciate that we can now change this microtubule array to this banded pattern instead. And actually, we can now take a lot of like different tricks in terms of bioimaging analysis tricks. And basically what we're doing here is, in this movie that you see over here, we take two next door images to each other and we overlay them on top of each other. And then we subtract those images from each other. And that's basically what you're now seeing, hopefully, in these channels. So these are growing ends of microtubules, and these are shrinking ends. And you can hopefully like visualize or like see that over here as well. And so we now measured many different aspects of how the microtubules were behaving. And the thing here was to try to recapitulate this in a model. And so that model, we basically like made this in a computer, right? Where you have the cylinder that we populated with sticks. And those sticks are behaving like the microtubules. And I hope you can appreciate that over time, we can begin to recapitulate like how these patterns are emerging. takes a little bit of time. I'm sorry. So you can now see that we're beginning to form these bands also in the model, right? And so we think that this is a really neat way of combining basically biology with modeling, right? And so basically by changing or tweaking our model, we can now begin to see what do we need to readjust for this microtubule behavior to make different types of patterns. And of course, this has some biological relevance as well, because we have all these different patterns of secondary cell walls that we now potentially can explore. Right. So we think that this is giving us some form of possibility to tweak these like cell wall patterns, and therefore also tweak perhaps how these secondary cell walls are made. So this part of the talk was basically to understand how wood is patterned and potentially like also give us a way into making new type of patterns in secondary cell walls. So you might wonder, okay, you have only looked at these like microtubules, right? So what's happening with this like cellulose synthesis during this time? And actually, this is a really interesting question, because we have one protein complex shown up here that is involved in making primary cell walls. But that is shifted out to another protein complex that is now making cellulose during secondary cell wall synthesis. And so our thinking here was that if we now have a marker for this primary wall complex up here, together with another marker for the secondary cell wall in different colors, and we cross that into this inducible line, we can now look at basically the transition between making a primary wall to a secondary wall in real time. And this actually turned out really nice. These are just stills from that, but anyway, I think that you get the picture. So you can see the primary wall is made, of course, before we are inducing. And then you can see that we slowly begin to form these bands, right? And there you can also see that you're slowly now, the primary walls are disappearing, right? So you're not making any more primary cell walls. On the other hand, you can see that you're inducing now these secondary cell walls, and that you're again having these patterns. What's really neat is that actually you have an overlap between the primary wall synthesis and secondary cell wall synthesis. So now we can begin to look at like, okay, are these complexes actually moving in the same way, right? And so we quantified this, and we found out that actually the secondary cell wall complexes move twice as fast as the primary one. So that means that they're actually spitting out cellulose twice as fast during the secondary cell wall as the primary cell wall. So in a way, you can kind of imagine this as this like passing lanes on a highway, right? Where you would have like the primary wall assays probably moving in the slow file, and the faster secondary cell wall using complexes moving in the fast lane. All right, so these are kind of ways to visualize cellulose synthesis and pattern formation in woody cell in real time. So I'm going to talk a little bit more about microscopy as well. So maybe a little bit like along the line with what Mikael was doing, but we're actually not using so much these off-the-shelf company-based systems. So we're actually trying to modify the systems to suit our purposes in the lab. And so one system we built together with 3i, you can see here probably. So it's a regular spinning disc confocal microscope that Mikael was also talking about, but you can probably see that we, instead of having this horizontal stage, we actually now have a vertical stage instead. So in this case, all you do is like unscrew your objective down here. You put on this objective inverter, and then screw on your objective over here. And now you can image basically on this vertical stage. And for plant biology, that makes sense, right? I mean, plants are typically not growing in this direction. They tend to grow vertically. So you can now begin to image things in real time over longer periods of time. So many labs now have these like vertical stages as well. They don't need to be that expensive, actually. It's this was a really cheap solution to the system. But the thing is, like now, most of the imaging that we do on media plates. And we all know that plants do not grow in media out in nature, right? So in the lab, we're trying to get around this problem and actually like make small incubation chambers where we can grow plants directly in soil and to image them in soil, right? So we make small rhizotrones, if you want. So this is just like things that we've developed in the lab. So here you have an incubation chamber. You can see that there's soil, right? And you can now, for example, now follow this root throughout the soil the whole way, right? And you can now, of course, zoom in on any region of this root that you're interested in. You can, of course, also now begin to follow root growth in real time. And that's what we're doing here. For example, you can now see that you can really follow these roots nicely with a plasma membrane marker in soil, right? And so in the lab, we're quite interested in compacted soil conditions, right? So it's a major agricultural problem. So basically in the next one, you can see now a root growing in compacted soil. And you can hopefully see that the length of the cells are actually much shorter and the root is more swollen. And that's a typical response you're getting from compacted soil. And so of course we can take this further. What we've done now is basically combine different soil types in one chamber. So now you have compacted soil down here and non-compacted in the top. And we can begin to look at now how the root is behaving in real time when they're basically like hitting this compacted soil, right? And so this is just a postdoc here, and here's just like one root that we're following through this like compacted condition. So, as of course, we have only tapped the surface of what we can do with this system, I would say, right? So you of course can now begin to look at like how are the different cells behaving when they're hitting, for example, this compacted soil. How do different marker lines for like hormones and for different signaling components also behave during this process? And this is really important for cell biologists to understand, to actually understand how this system is reacting to these conditions. Right. And so we think that this can be used for a lot of different purposes. I mean, in terms of root microbe interactions, for example, different types of abiotic stress responses, and of course, understanding signaling in roots in real soil, or at least like as close as we're getting. We can perhaps try to like dig down a confocal microscope in soil, right? But I think this is probably the closest we're going to get. Right. So back to this confocal microscope that was shown before as well, right? So I know that this is pretty pricey. I mean, I think an investment for a new confocal microscope is pretty expensive. I think we paid 10 billion Uzbeki som, I think, for our system. It sounds like an insane amount of money to me. It's about 5 million Danish crowns. So that's more reasonable, I would say. But you don't need to buy these systems, right? You can build microscopes yourself as well. You just need to have the right people around. So in my lab, we have 3 physicists that are basically building microscopes themselves as well. And so this is one system, and it costs actually 0 crowns or 0 soms, because we could use material from other microscopes and just take those out and reassemble them into a new system. And so this system is quite unique because most of the stuff that we are doing in cell biology in plants is based around fluorescence. Right? So you use these fluorescent proteins, you use different dyes, or you use antibodies, right? But you and me, we don't use fluorescence, right? I mean, I know that that's Barno. I know that that's an empty chair. And we're using light interactions with surfaces. So it's light scattering and refraction and reflection, right? And we interpret this in a really, really good way. And so this microscope is basically set up to combine fluorescence microscopy in this light scattering microscopy. So basically, if you follow here, you have like basically light coming in, you have your sample up here, and then we basically divide the signal into either the fluorescence, whatever wavelength we're interested in, or actually detecting the scattering light. And so this is just a surface of a leaf, and this is a single cell here. Basically, these epidermal pavement cells of Arabidopsis. And this is with this light scattering channel. And I hope you can appreciate that there's a lot of movement of different stuff in here, right? And this is basically capturing all organelles and bigger vesicles. So the resolution is about 150 nanometers. in real time in these cells, right? This is kind of cool because there's no bleaching now, because we have no fluorescence. So you can image this for days if you want, right? Problem here is we don't know what the heck this is or this is, right? So that's why we need this fluorescent channel to be able to pinpoint down what are those different organelles or vesicles, right? But of course, over time, we will be able to track these, and I hope you can appreciate that we're tracking here now different organelles and vesicles in root hairs in this case, right? But we can learn from this. We have basically AI and machine learning tools that we now can begin to tell, okay, this is behaving in this way. And over time, we believe that we can actually use this to say, without the fluorescent channel, this is this and this is that, right? And we think that this is opening up like a completely new field in terms of doing cell biology in crops, right? We all know how difficult it is to actually do cell biology in any types of crops. And I think this might be a way around this. So what can this be used for? So we can image, of course, vesicle and organelles in crop plants and other organisms. We think that we can use this, actually, we are pretty sure that we can use this. We're doing it currently for infection, for example, and plant pathogen in barley, and of course, any other crops as well. And we think that we also can use this to couple to the soil chambers that I was just showing you as well, to actually get like a better view of non-fluorescing, I mean, basically non-cell biology tractable systems, to actually look at those in real time as well. Finally, actually, so these are some of the people. We have this experimental microscopy lab. So we're actually building, I mean, not only this microscope, we're actually building microscopes where you can look at mechanical stresses in tissues and cells, super resolution microscopy systems as well. Most of those cost a fraction of what a normal microscope would cost that you buy from a company. The last thing, I didn't really know what people might be interested in this context, but we're also using something called optical tweezers, where you capture you can capture just like a vesicle or compartment or whatever using a pretty high powered laser. And what you can do is, well, you can do a lot of things. You can actually measure forces between proteins with the system. But in this case, we have used it to fuse protoplast and vesicles. So you can basically now introduce new material into a protoplast by just fusing the vesicle to it, right? And so here is just like actually in this case, we're fusing two protoplasts together, which I don't know what the purpose of that actually will be, right? But we can do that. But of course, you can now begin to introduce new proteins, new type of lipid environments into protoplast and see how your proteins of interest are reacting to that. So with that, I of course want to thank the lab that you see here in the back. Actually, most of the work that I was showing today was done by René and Arun. Arun is now a group leader at the Max Planck Institute, and René has an emenuta at the University of Potsdam, so they were fairly successful afterwards. And then we have collaborators for these specific projects, and of course also funding organizations that made this work possible. So thank you very much, and I'm happy to take any questions.
Yeah. Thanks so much, Stefan, for the wonderful talk. So yeah, we can see that this microscopy tool is really powerful, that they can show us really wonderful view, things happening in this cell, so which we cannot see by our eyes, right? Naked eyes. Yeah, then now time to discuss.
It's the microscopy mafia. Right.
Bad questions. No, thanks a lot for a really impressive talk. And I really appreciate that you mentioned that you don't always have to buy the fancy stuff. If you do it self-made, anyway, it's better because it's adjusted exactly what you want. Two questions. One is about the little rhizotrope. Because I mean, nevertheless, you have to have the surface where you can image.
Of course.
So that makes a difference also.
Of course makes a difference.
Because otherwise Blue Root would take another
Sure.
way, probably.
I-
But
I- yeah, I would still argue that it's better system than the
Yeah.
media, basically. It's-
Exactly. Yeah. And finally, one question about the optical tweezers, because there it's similar. You use it only for protoplasts,
Currently, we're using it for protoplasts and different sized vesicles. Yeah.
because I think also for the cell, if it has a cell wall or not, makes also a big difference.
Yeah, of course. In this case, we're interested in introducing, for example, a new sterile environment and to see whether proteins that we are interested in actually goes to that environment or not. But you're right. Of course, any type of experiments that you're doing on protoplast have to take that very careful. It's not a plant.
Yep.
Right. Thank you.
Thank you for a talk. I'm also want to ask about the protoplast because we're working on the system, and we had quite a problem to isolate the protoplasts from woody plants. So actually, we are struggling a lot isolating protoplasts from tea, Camellia sinensis, and it can probably be related to the cell wall structure. And this is actually what would you suggest to do to make it simple? Because we just take at the end we take the younger leaves ever possible, and we have some yield. But as you showed here, it is possible somehow to increase the woody cell type formation. Are there any ways to decrease them or somehow treat woody plants in tissue culture to make protoplasts more available?
It's a good question. Yeah. We are struggling ourselves actually also to make protoplasts from certain species. We're working a lot with red macroalgae now, and making protoplasts for those has not been trivial. So it's a really good question. In terms of, I mean, I guess you can use tissue cultures, right, to try to get more cells that don't have the secondary cell walls, and maybe that will help you to get more protoplasts out from it as well. I don't know.
It's other way around. So actually, the
It's
colors,
other-
yeah.
I'm sorry. I'm not sure that I will be the best person to help you there. Sorry.
Maybe I missed. So you overexpress this factor which made secondary wall in all tissues, and maybe I missed this. What phenotype did you get?
Oh, it's a terrible end. The seedlings die, because they're all dead cells, right? So the whole seedling just turns yellow and dies. So in terms of phenotype, it's a rather boring phenotype, I would say. Yeah.
Did you try to knock out this factor?
If you knock it out,
Mm.
or you cannot knock it out. I mean, I think the seedlings will die very, very early because they're unable to transport water and they're unable to stand up. So that would also not be a good end, I think. So.
May I ask a quick question about the same? So you showed that the primary cell wall stops producing, secondary starts producing. Does it mean that after this moment the cell cannot move anymore, grow or bend to the side?
Yeah, so there is some flexibility left in it. So when you make these they're called protoxylem, so it's basically the first forming xylem, then these banded patterns actually allows the cell to expand a little bit, right? But when you make these pitted patterns or the holes, then it's impossible to expand anymore. You can probably think about that as well logically, that if you have just like holes, then expanding that cell wall will be extremely difficult, right? So typically, like the protoxylem formation, you have some expansion, but the cells ultimately die anyway, right? So there's a programmed cell death program that is kicked in.
So it means the secondary cell wall formation is somehow related to the programmed cell death.
To what?
To the programmed cell death afterwards.
That is the end of that process, yes.
Thank you.
Thank you so much for your interesting information. I would like to know the difference molecular density of synthesized cell walls, because as you know, asalination is one issue in global change, climate change. And this reason, plants lose forcibly water content because of osmotic pressure in saline condition. In this reason, I would like to know how synthesized cell wall reduce pressure, osmotic pressure in saline conditions. Thanks so much.
So it's I mean, in terms of osmotic pressure from the inside, you mean in that case, or?
Yeah, inside.
Right. I mean, so the vacuole is taking up water that is pressing against the cell, and that actually leads to typically more cell wall synthesis and expansion of the walls. So that's basically like how plant cells grow, right? So I'm not sure if that's answering your question or not, though.
Thanks.
Okay.
Sorular var mı? Bu mesela hazır coffee break bizde. Thanks so much, Stefan, for the nice talk. Now we have coffee break, and after the coffee break, we have two more presentations. Then we close the workshop today. So see you later. Madam. In the kitchen. So we are continuing our section. Now again, Michelle Melzer giving continuing of this formal presentation. So Michelle? Yeah. Yeah, stage is yours, please.
Okay, so thank you for being here again. You're tough. So the next session of microscopy I will talk now a little bit about how to use and combine different methods in selected research topics. And I will show you first, before I will start with the research topics, I have added 3 slides which didn't show up in the first talk. So hopefully now they will do. So I will give you a just brief introduction about history of microscopy. So how does it start? It's starting with the Romans and Greeks very, very long time ago, as they were using burning glasses. So a little bit later, Seneca and Pliny the older one discovers in a ball with water that there is a kind of magnification. So then a couple of years later, Abu al-Hasan Haytham, he's noticed as the father of optics, and he was the inventor of the magnifying glasses. Later on, Galileo, who is doing a lot of things, was also involved in microscopy. So because he constructs the real first microscope, the so-called Occhiolino. And then a little bit later, Robert Hooke came onto the stage, and he writes the first microscope reference book, Micrographia. And you will see that it's a lot of with imagination what he has seen. So this is a sperm cell, which has already a body and a slight face. And the other thing, you see also a smiling cricket. So that's what he observed. But nevertheless, I think it was amazing what they have shown these days. Okay, not all of this slides. So unfortunately, there is again not showing all the slides because I would like that it's going on with the invention of the electrons. And finally, there were 2 Nobel Prizes in chemistry, but it was for microscopy. And this was Stefan Hell from Göttingen University about STET microscopy, which is about confocal microscopy in a special way. And then 3 years later, in 2017, it was Jacques Bouchard and 2 other guys who got the Nobel Prize for cryo-electron microscopy. So the first thing I will talk about is the use of genetic diversity to use reduced grain shattering, which is a problem in breeding. And this was a project between our group, then Satellite Collection North of the IPK, and then GeneXpro, which were responsible for transcriptomic analysis, and the breeders in Steinach. So what was the plan? The first thing was to genotype accessions with firm and loose seed retention, then screening them, sending back the material. And then what we are doing is the analysis of the so-called adcision zone, which is responsible for seed retention. And we did a lot of morphology and histology. And in the same time, we did a screening of accession lines from the gene bank. And finally, we take care of the segregation populations. And finally, the idea was to do again histology, then gene expression analysis, and DNA marker development. And the final aim is to invent new breeding lines for the breeders. So what we did, we have used the on of a perennial grass, Lolium temulentum. What you can see is a vibratome section, and you see the floret. And the interesting part for us after the first, okay, where you are, okay, here it is probably exactly this area here. Here you have the development Caryopsis, and we checked them at different developmental stages. So the question where is the abscission zone located, because it's not known. Then are there structural differences between high and low shattering genotypes, what one can expect? And the dream was just to develop a quick test that you go into the field, spray something, and then you will see, okay, shattering resistance or not. So the starting point was the analysis of 19 genotypes and with high and low shattering. And this was used then for histology work. We did some serial sectioning of in a histological way. We created a lot of kind of videos, which was supported then by Felix Rose and Stefan Ortleb. So at different little stages. So, and we tried to identify the abscission zone. So we checked also different floret, different parts here. And then what we did is just cutting in this direction into the Caryopsis, and what you can see here, this is a genotype with a high shattering. So this is how it goes. The first video shows a journey from here into the Caryopsis. What you can see, we are still here in this part, and then we are moving into the slowly into the Caryopsis, and finally what you can see are the pollen and in the anthers. So this was one part of the work, and on the other hand we did some serial section here in a longitudinal version and with the same plant. And the interesting part for us was then finally what you see here in the red square. And here you closely, you see something here going on, and the idea was really now to identify the structure of the abscission zone. So then the next years, we did some screening in the field, and we selected a few interesting genotypes with the most contrasting lines. And then we did also some X-ray analysis because this was much faster. And this was again in cooperation with the Fraunhofer Institute in Würzburg. And on the same time, we used exactly a transcriptome analysis from the part that was here is shown in the square. And this is how it looks like for X-ray microscopy. And then what you will see on the left-hand panel is a genotype with high seed retention. And what you can see that the cells are really connected to each other, so it's really tough. So therefore they still stay at the ons and will not lose it so far. On the other hand, you see a genotype with low seed retention, and you will immediately see then the differences because the abscission zone, the two layers, are loosely connected to each other, and therefore there is a low seed retention. So what we have found is we identified the abscission zone itself, and then we have shown that they are loosely connected in 2 or 3 cell layers. Then because the cells are in the resistant genotypes are much more connected, and in the tall meadow oat, I didn't show any images about that. Here it was very difficult to show, and we failed to really show it where the abscission zone is located. And finally, we were not able to do a quick test because
the abscission zone is really surrounded by so many tissue that it's not possible. So the transcript analysis, it's an old slide. It says it's still in progress, but we didn't get really nice results out of it because unfortunately, the person who did the work at that time, he passed away due to disease, and therefore it gets a little bit stuck. So we don't know exactly about the genes so far. But in the meanwhile, the Saatzucht Steinert, the breeding company, are using the results to create new genotypes. So the second part of my talk is about the investigation of the role of strigolactones in response to drought stress. And this is a project which is done together with Jochen Kumlehn and Irene Fontana. She was working in both groups and she's still working in Jochen's lab. And it was an initiative to have a cooperation with the Polish group. And Gerd Sensel also, he was at that time when we started the project, he was still at the IPK and then moved to Düsseldorf. And finally, our Polish partner, Marek Mazec from the University of Silesia in Poland. So I just briefly go through a little bit about the strigolactones. They are regulating processes in ex planta. And okay. So strigolactones are involved in drought response, and this is known for Arabidopsis, rice, and tomato. And it's an increased tolerance to abiotic stresses. So, and the question is now, strigolactones are also involved in drought response in barley. So the idea was to generate barley mutants in the strigolactone biosynthesis on one hand, and also in the signaling pathway. And so the Polish group had the tilling mutants, and Irene Fontana in Jochen's lab created all the CRISPR-Cas lines, and they were both used then for further investigation. And I will show you just one example, and this is about D14, which is a part of the signaling pathway. So here you will see how it looks like after drought stress, and the drought stress was given 14 days. And you will see that the D14 mutant of course suffers from drought much more than the wild type, and which, oh, okay, there's something happens here. So there is a lower water uptake, and there is higher stomata conductance and impaired photosynthesis. And then we checked also morphological parts, and you see a stronger alteration of chloroplast structure here. Here you have the control of the wild type, and here, and you see that the mutant looks really, really unhappy, I would say. Then there is reduced thickening of the epidermal cell wall, what we can expect here, and it's less pronounced in the mutant. On the other hand, also, the cell wall thickness is less, and also the cuticle thickness is reduced, and therefore they suffer more from drought stress. So then, of course, there was also transcriptome analysis, and we discovered 406 differentially expressed proteins, and there were a couple of different expressed genes and 17 expressed transcription factors, which are expressed in the wild type but not in the mutant. And a couple of these data have also published in the meanwhile. Then there were also a hormone measurement, which shows there's a downregulation of phthalic and jasmonic acid, and an upregulation of abscisic acid and cytokinins as well. So then we checked also an effect on the wax layer, because this is also important in drought stress. And what you can see, there are two things. So in the wild type, after drought stress, it's much denser than it is in the mutant, and also the structure is
different. In the mutant, it's much more tinier, and also the structure itself of these plates looks a little bit different. So we were able to identify extracellular tones, dependent regulatory mechanisms. And as I told you that there is 17 transcription factors differentially expressed. So the next one, this is a main topic at the moment for us. And this is Twan Rutten of our group. And he did really tremendous work in the Rachis of Bali with a focus on the vascular organization. And this is done with two groups. It's Thorsten Schnorbusch, who started to asking us, can you please check the vascular tissue? And this was maybe 6 years ago. And in the meanwhile, this is a very, very strong cooperation with a lot of students from his group. And also here he was working, Yongyu Huang, he was working also in that group and now has his own research group. And we still continue working with them on this topic. I just show you now how it goes. And of course, you will see some animations and reconstructions as this, and all of them has been done by Stefan and Felix. So what we are used in this project is really the whole spectrum of our microscopes. We did some histological staining to stain the vascular tissue in a histological manner. Then we did fluorescence microscopy. Then, of course, morphology analysis and the SEM at the different developmental stages, serial sectioning again, and 3D modeling, and of course, ultrastructure analysis. So here you see the spike development of Bowman, and this was imaged by digital microscopes. And what you can see here on the right-hand side, that here it starts the abortion of the apical florets, and finally they are gone in the mature plant. And of course, it would be helpful if we can control that so that it will not happen in such a way, and this means that you have more yield at the end. But we have to know why is it the case and what is the mechanism behind it. So just a little bit more in detail, what you can see here at stage 5, it looks more or less happy, the apical part, and it starts at the Waddington stage 7 around, where you can clearly see that the shape of the apical part is changing. And we would like to be interested much more how it looks like. Then we did propylium iodide staining here in the wild type, and you can see this is a stain to show apoptosis, and it was clearly in the upper part where it starts. And then we checked it also in the SEM, and here you can see the vital wild type, and here it starts already with the degeneration process in the apical part. So a little bit more in detail about the tip region. So we are going step by step. We're starting at the very early stages. You see the apical meristem, then the transition of the apex. Then there is a so-called double ridge stage, which you can see here. Then early triple mount stage. Then you have the early glume primordia showing up. And then at roughly around 3, you have the early lemma primordia, then fully developed lemma primordia, then you have the anther primordia, the pistil showing up, and the horn primordia, and then at the white anther stage more developed. So then we checked out maybe what would be an interesting stage, and we decided to go for stage 8, where there are a lot of changes. And the idea is to check it more in detail. How is the organization of the vascular tissue? So we did again serial sectioning, and then what you can see on the left-hand side is how we did it. These are about 500 sections that were put together. And then we were able to assign the different tissue. So in red, the lateral veins, then the third order median veins in pink, then the first order median, the glums, the second order median vascular tissue, and the vascular tissue from the lemma, and this was combined. And then we made a 3D model. And what you can see here is the different parts of the vascular tissue combined and in a single. You have the laterals, the median second and first order, then the median last order, and then we put it together again into our model. And this is what you can see here now, like a Christmas tree, where you can really see the connections. Nevertheless, we did it now because this was a side view, just to check what happens if there are different vascular veins fusing to each other. And therefore, we now see a movie starting from the top, going to the base. And if you check maybe the red one, which is the lateral veins from the rachis, and just to check out if there is any fusion during the whole video. So it's starting from the top, then the main lateral root appears, and then you see the glums coming all the time, mainly fusing with the first order medians, and later on also the lemma is coming into and they are fusing with the second order medians. But so far, nothing happens with the lateral ones. So they are just staying by themselves. And this is going really until the end. And I think this was very cool because you have the point of view from the top to the base, and before we had it from the side. And this is how it looks like in the pistil then. So we have shown that spikelets are attached to and supplied by the central part of the rachis, and that the spikelet veins are unable to attach to the lateral rachis veins. And so this means that the transport and delivery is spatially separated in the rachis. So the question, does this organization change at lateral developmental changes, maybe due to brain filling? And therefore, we also checked a little bit later stages. And we would like to check out a little bit more in detail what happens in the nodes, just to checking out how is the organization there. And this was done again by confocal work, and you can see now how it goes from the base to the top, how it looks like. And you see also changes in the vascular tissue if pay a little bit attention. And so the idea we picked up, because it's a lot of work, so you cannot do everything in a week or so. It takes a while, and we pointed out something in the center, and you see on the right-hand side. And then you see how it goes. So this is a segment just showing how it looks like. This is in the node, and you see that branching to the florets. And then the question was, how is the organization of the vascular tissue? So we did tissue assignment, and this was again done by Tuan Rutten. He's sitting days and days and just making colors to the structures, about 250 sections. And again, tissue assignments for the palea, glums, rachilla, and so on. And then I will show you how we build it up. So we're going step by step. First, the median veins, then the lateral ones, then it comes with it to the peripheral veins, then other outer peripheral veins, then some more veins, the glums, the palea, and the vascular tissue of the rachillas, of the ansa, xylem, and other answers, and finally the radicals and the lemmas. And at the end, lateral spikelets. And this is a model, finally, which gives you a lot of information about the connections from the rachis to the spikelets. Okay, then he did a lot of other analysis. For example, he compared the tissue area to the internode number, and then the vascular area and the number of the major veins. He just did a lot of measurements here, and finally he published the work with the conclusion that the rachis diameter and the vascular area, they are really strongly correlated to the spike size, while the vascular number is really weakly correlated to the spike size. And this was published recently last year. So then another thing was that we would like to know also about the organization of the vascular tissue in the internodes. And this is a common project with Ricardo Gil and Yongyu Huang, and also with the KWS breeding company. And Bakhtiar Rahmanov, he did really tremendously work because he was preparing 200 genotypes, and this was very difficult material because this was mature tissue, which is very hard, and he really is an an expert for that, and therefore I would like to thank Bakhtiar a lot for this work. And this is just to show you about the connections and the combinations what we have at the IPK. You see it's a real network of the different microscopy techniques, which makes it really very powerful because they are in one group. And then connected with a super resolution and the light sheet microscopy, I think it's a really, really well-equipped microscopy platform. And therefore, this means that we have a lot of cooperations with the Institute, as you can see, that nearly with all the other groups. And finally, I would like to stop here with this really beautiful image. And this was done by our trainee, so just for fun at the end of the day, and it's a really amazing image. And therefore, I would like to thank you for your attention.
Yeah, thanks so much, Mitchell, for the really great talk. It's really amazing. I speak in my language. Bu mikroskopda ishlarni ko'rib, endi to'g'ri men bular bilan birgalikda ishlab, bularni o'sha ishlarni qilayotganimni ko'rganman. Masalan, har doim nima olardim-da, endi masalan, qanaqa qilish mumkin bunaqa qilib? Endi bu narsalarni har doim bizada yo'qligi, O'zbekistonda bu narsalar bizga qanaqa qilib biz olib kelsak bo'ladi, O'zbekistonda mikroskopni qanaqa qilib rivojlantirsak bo'ladi degan narsa o'taveradi-da. Lekin hozir ko'rib yana o'zim bo'lmasa ko'rganman, yana hayajon, yana bir nima desam, til bilan tasvirlay olmayman. Kimdadir savollar bormi? Masalan, o'zbek tilida o'zinglar qanaqa taassurot qolganinglar kimdir gapirib bera oladimi? Keyin anaqa bemalol fikrlaringni bildirishinglar mumkin.
Thank you very much. Is it the same approach you demonstrated for the veins as Barno also showed in her lecture about this vascular tissue annotation in the series of imaging?
Yeah.
And so it looks like this library to train the artificial network. Are you going to try to avoid this big work by trying to train this artificial intelligence for this not to make it
Yeah, sure.
yeah.
That's exactly what's coming up. So I joined the Gordon Research Conference this spring, and it was absolutely totally amazing what's going on with image analysis and reconstruction software based on AI. This is really, since the last conference, a super jump. And so I'm in contact already with some people because I would like to use exactly the same image stacks and then just to see how it goes with AI.
Yeah, it's really interesting whether you can just substitute
Because
this huge work by
Yes.
AI. Yeah. Okay.
Yeah, it was. He was really sitting for really days and days and days, but finally, the outcome is really amazing.
Thank you. Yeah, that's actually a related question. So I guess the segmentation is now made by hand. Huh? But I mean, do you think that it can be automated? Because I guess the cells look relatively similar across a section,
Exactly.
right? So any ideas for how you can actually do that in an automated way?
Yeah, this is exactly the point I would like to figure out with those guys for this automatic, they say it, automatic segmentation. Because Tuan, he told me very often that he thought it's that structure, and then he realized, no, it's the wrong way, and then it's going back and so on. And that's the question if AI will also recognize it. It needs a lot of training, I assume, but this is exactly what we try to figure out. But I think in the future it would be, of course, time consuming. I'm not sure whether it's always precise, because with your eyes, you see sometimes things a little bit earlier. So, but the combination maybe is the perfect thing.
Yeah, o'zbek aniq fikringlar nimalarni olganinglar gapirib qiziqda.
Thank you very much, Michelle, for your good talk. I express my opinion in Uzbek. Barnoy will translate. To'g'risi, mana Phenomics degan soha rivojlanganda, umuman dunyoda Phenomics, ya'ni bu mana shu suratga olib o'simlikni butun morfologiyasini rasmlar orqali tahlillaydi. Bitta shunaqa dasturlar bor, shunaqa uskunalar bor, o'nlab, 20lab yoki 30-40ta belgini screening qiladi bittada. Nafaqat uni O'zbekistonga olib kirish kerak. Hozirgi tadqiqotlarni ko'rib turibmizki, nafaqat bu Phenomics, balki butun o'simlikni ma'lum bir organlarini anatomik tahlilini qilgan, strukturasini qilgan. Bu Barnopa, siz nafaqat to'g'ri qayg'uryapsiz, hozir O'zbekistonga buni olib kelish kerak, bunaqa texnologiyani, mikroskopiyani deb. Nafaqat texnologiyani, umuman kadrlar salohiyatini ham shu jihatdan oshirish juda muhim, chunki kadrlar salohiyatsiz bu ishlarni qilib bo'lmaydi. Bizda ham bor mikroskopiya bilan shug'ullanadiganlar, olimlar, ya'ni hujayrani genom darajasida, xromosoma darajasida tahlil qilishadi, lekin bunaqa strukturaviy jihatdan o'simlik organlarini qilish, I'm in shock. Juda ham yuksak darajadagi tadqiqotlar. Katta rahmat shunaqa tajribalaringizni biz bilan o'sha o'rtoqlashganingiz uchun.
It was a long question.
No, it was not a question. Just he just sharing his feelings so that we can also bring this technology to our country and also the experts together. Because if the technology is there, like without experts, like we can't do anything. But they're like also a new, I don't know, it's not new, like Phenomics kind of research field. They're really describing the morphology of each trait or each plant in a good way. So they're like really amazing. And also the in organelle level, cell level. Yeah, it's really wonderful.
I think it would be great to increase the cooperation.
Yeah,
I think it's very helpful if you combine all these techniques with the genomics and proteomics and whatever. And this is exactly what is going on more and more, that you have these combinations. And I think, as I tried to explain in the first talk, is that you don't always need the really, really fancy stuff. I mean, it's time consuming, but as I showed you, you can get it also with simple, not really simple, but with techniques that maybe you can make within the next years, just to have some basic things, and then maybe you can build it up step by step. I think I hope that it's possible, and hopefully we can support you there.
Wonderful. Yeah, during my PhD, I had also a little part of task where I have to do cytology or histology, and actually, at the beginning, I didn't want to do because my time is running out.
I remember that.
Yeah, I want to finish really, and I do not want. But my supervisor oh, he's not here now. He was like kind of encouraging me, Bano, it's like really interesting field. You will learn the things and just be passionate. You will be finishing your PhD. And then now I did my histology by helping Michel and Twan, and I learned, and I now proud of that. I know about the microscopy things, like it's really amazing.
And also, Bachtiar did really a lot of work. He was 9 months, and I think it's always the best if you don't do the easy sample, but the really bad one, because then you learn much more. And I think he's quite keen to do the basic things here.
Yeah. Good. Thanks so much, Michel, for the wonderful talk again. Our last speaker, again, a last talk, not last speaker. Stefan Persson. Stage is yours. Oh, su var mı? Su'daki kimlerdir su? Mesela skoçya çikili buna su. Ha? Yok, içilmegen. Kimlerdir o kendilik içilmegen var mı? Yok, içilmegen.
How about a word like I hope this is not my presentation. All right, same title. It's a great title, so I changed it, right? Yeah, I want to, of course, I'm the one standing in between you and a delicious lunch. It's been really good food here, so I'm sure I have to speed up a little bit for you to actually get there, right? Yeah, we're going to talk a little bit more about cell wall synthesis. I did a lot of microscopy bits in the first talk. We're going to talk a little bit more about molecular biology, perhaps, in this talk. Just a reminder, this is basically how the cell wall look like, right? Remember this jungle of mainly sugar-based polymers, right? Coming together around each and every plant cell. I showed you these questions, basically what we're interested in my group. We're going to talk about two other aspects of cell wall biosynthesis or modifications in this talk. Not going to focus on development, but rather environment, more specifically salt stress, which also is an important problem in agriculture. I'm also going to talk a little bit about how proteins interact in cellulose biosynthesis, and also hopefully introduce a methodology that might be useful to some of you to understand protein interactions. All right, I showed you this as well. Yeah, it's a pretty cool movie, I think, in terms of following cellulose synthesis in real time, right? Let's see. And this is basically then how the cellulose synthase complex look like. So you have this central part that contains these cellulose synthase proteins, right? And then you have some additional proteins here that are contributing to cellulose synthesis. And we'll talk a little bit about how we come up with those proteins or the corresponding genes as well. And actually, as a backdrop to this whole story, you can see here that this protein complex actually consists of 3 different main components. They're called cellulose synthase 1, 3, and 6 in Arabidopsis. Right, so intuitively, these 3 proteins need to be present in the same cell at the same time to form a complex, right? So if we now take a step back, the genes that encode these proteins would probably be expressed in a similar way as well, right? So if one gene was on, the other 2 genes would probably also be on. And actually, this goes back again to where I overlapped with Sue at Carnegie. We did these co-expression analysis at the time. So it was actually some of the first co-expression analysis, I think, in plants. But anyway, you can see the expression patterns of these 3 genes across many different conditions and tissues and organs and so forth, right? And you can see that they're really highly correlated with each other. So that means that these guys are co-expressed with each other. Now, if you now take the next step, if there will be other genes that have the same expression profile, we would assume that they maybe are connected to cellulose synthesis as well, right? And basically what you can do here is, of course, identify those expression profiles. Cool. Now I have plenty of water. And then basically order knockout lines for those in Arabidopsis. That's really easy, right? And just assess whether they're impaired or have defects in cellulose synthesis. Right? Another way, I mean, of course, we can overlay expression profile after expression profile here and look at them, right? But another way of doing this or showing this is through these like ginormous networks, right? Or hairballs, if you want, right? So in this network, you would have nodes that represent a gene, and then you would have edges or lines between 2 genes that then represent a certain co-expressed relationship. And of course, I've zoomed in this ginormous hairball for our SES-A genes, and they're really close in this network. You can see them together here, 1, 3, and 6, right? And so throughout the last 15 years or so, we have basically like identified many genes that are now really important for cellulose synthesis through this approach. So these are genes that encode proteins that are involved in actually binding to cellulose microfibrils. There are other ones that are basically associating the SES-A complex with the cytoskeletal components, and yet other ones that are involved in basically putting together the complex and signaling. So in this talk, I'm just going to nerd out a little bit on the mechanism of one of these. And it's basically following maybe Brandes' talk a little bit, like going a little bit excessive in that direction, I would say. But I think it's important to understand like how we're getting from a protein of complete unknown function to delivering an exact mechanism for how this protein works in the context of cellulose synthesis. So to be, okay, actually we need to know as well that the SES-As are connected to microtubules, but I showed that in the first talk already, so that's fine. So in a first study, we briefly outlined a crude mechanism for how this protein works. And basically what you can show here in blue is that this protein, companion of cellulose synthase, maybe not the most original name ever, but it's basically a companion of the SES-A complex. It's hanging out with the SES-A complex, moving with it when cellulose is synthesized. The function of this protein is not becoming present in a sense until we expose plants to salt. And so what's happening during the salt stress is that the SES-A complex is removed from the plasma membrane inside of small vesicles, and the microtubules are falling apart. And this protein is now able to reassemble the microtubules, and therefore this complex can now go back to the plasma membrane and continue to synthesize cellulose, okay? And this is basically a way for the plants to maintain directed growth during salt stress. And so obviously now in the absence of these blue guys, it's a double mutant of the 2 main homologs of these, you can now see again the SES-As are internalized, microtubules falls apart, but this protein that is supposed to like put together the microtubules now is absent, and therefore no more cellulose is being produced. So this is, of course, like a crude mechanism for how this protein works in the process, right? But we were really interested in the kind of molecular mechanisms behind this. How are the proteins actually doing this, right? And so we focused on this inside part of the protein. So this is basically the part of the protein that is facing the cytosol and interacting with these microtubules. And the reason for that is that by only taking this piece of the protein and putting it back into the double mutant, we can restore growth on this salt-containing medium. So basically what we do here is grow seedlings for 3 days and then transferring the seedlings to salt. And then you can see that the wild type is continuing to grow, but this mutant is not. But if you now just add this piece, you rescue this phenotype, right? So therefore, we were really interested in how does this part of the protein actually work, right? So we did a lot of assays in vitro initially. So we expressed this part of the protein with a his tag. You can see it's produced quite nicely here. And then what we did was to incubate this protein part with microtubules. And basically, I thought this image was really amazing the first time the PhD student showed me, because here is an electron microscopy image, as Mikael has shown you before as well, right? But here is 2 neighboring microtubules and our protein that is now labeled with gold conjugated antibodies against this his tag. You can see that it's like in between neighboring microtubules, equal distance, right? So it's basically this protein is like gluing together neighboring microtubules, right? And so just to go back to your cell biology 101, microtubules are small tubes, microtubes, right? And you can see those here, and you can see that they're made of rows of tubulin proteins that come together to form this tube. And so now we wanted to ask, okay, if these guys are gluing together microtubules, where on the microtubule surface are they actually binding? So we did something called a chemical cross-linking experiment. So we basically incubated microtubules with our protein, and then we added a chemical that basically covalently link your protein with the microtubules. And then you can identify these crossovers via mass spec technology. And then we can map down that actually our proteins are binding in this red and orange configurations in between neighboring protofilaments on these microtubules. Right? So now we have like a pretty nice initial mechanism for how the protein engages with the microtubules. Right? So we have this CC proteins that aid in this gluing of microtubules and bind neighboring microtubules in between these protofilaments or these rows of tubulins. So how does the protein do that? So for this, we turn to NMR, another method I think you introduced briefly as well, Misha, right? I mean, it's basically NMR. So you label your protein and then you take magnetic pulses that basically like change the directionality or like the movement of your of the amino acids basically in your protein, and each amino acid movement is basically corresponding to a peak in this graph, more or less. And so our prediction of this protein was that it was unstructured. And actually, I'm not an NMR specialist by any means. You probably heard that from the explanation. But anyway, this peak distribution confirmed that the protein is unstructured. So this was nice, but not very helpful. But what you can do now is add microtubules to this. And then you would change the chemical environment of the amino acids that are close to the microtubules. So basically what you now do is like you have your NMR graph without microtubules and with microtubules. And you can see, for example, here, you have a displacement of the peaks. And that means that those amino acids are close to the microtubules. So you can begin to see what part of your protein is actually binding to these microtubules. And now we can nerd out even more, right? And begin to like plot basically the plus minus microtubule signals, and then look at which amino acids are now really in close vicinity to these microtubules, right? And you can basically plot this ratio and then find out that probably you have these 4 microtubule binding domain of a protein. All right, so that gives us basically like this type of image here, right? Where you have the SA complex, you have this like CC protein that now, basically like tethers two microtubules together through these potential 4 microtubule binding domains. And actually through more specialized NMR, you can actually nail down which amino acids in those domains are really key to that. And we mutated 2 of those sites and show that indeed those are very, very important for this interaction to happen. Great. Now we have the next step, right? I said we would nerd out on mechanism, and I'm not kidding, right? So the CC1 and 2, they are SESA-associated proteins that maintain microtubule coherence during salt exposure. But how does this relate to salt stress? That's, of course, the next question, right? So key to the next step of this project was basically to find interactors of our CC protein, because we thought that maybe there's additional proteins that are modifying our CC1 in some way to actually like change this during salt stress, or like the interaction during salt stress. And so we did this by simply taking a GFP fusion of our protein and then pulled that down with GFP antibody. And of course, we used a control for this. And then here is just like basically the overlap of positive interactors in response to salt. So we have 0 hours, 2 hours, and 12 hours. And here we were mainly interested in actually the overlap initially between those. So we have 26 proteins to choose from. One of those were Veronia. Maybe it doesn't tell you too much, but in a molecular biology audience, people will be like, whoa, okay. It's basically like auxin. I mean, it's involved in everything. It's one of those receptor-like kinase that if you do anything to a cell, this guy will be associated with it, right? So initially I told the student, like, let's stay away from this because there's so many groups working on this receptor-like kinase, right? But then I talked to a collaborator on another project at the stress center in Shanghai, Chun Chao Chao, and he told me that they also had come to a similar conclusion that Veronia and CC have some relationship. So we continued to work on this project with him. And so the reason why we were also like, okay, Veronia could actually have a function in this, because Veronia, among other things, actually like sensing salts, at least like the cell wall damage that is caused by salt, and then signaling in some way to get a response from the cell to this treatment. Also, you can see if you have Veronia mutant, so where you knocked out this Veronia gene, they're much less tolerant to salt stress than a wild type. So there were some interesting connections, at least. So it's not completely off. So Veronia is a receptor-like kinase. As Brande pointed out, usually you would have like a kinase substrate in that case. So something that gets phosphorylated. Potentially our CC1 protein, because it interacts with this Veronia. So the next question was, can CC1 be phosphorylated? Right, that seems to be an obvious next step. Yes, there's actually 12 potential phosphorylation sites. We confirmed many of them ourselves in phosphoproteomic analysis that can be phosphorylated. So that was also a nice thing to show, right? And so then the PhD student and our collaborators went further and confirmed the interaction with the typical like different toolboxes that are available. So we could confirm that the CC1 is interacting with this receptor-like kinase. Okay, so just to remind you, if we're lacking this CC1 protein, we have this internalization of the SESA complex, microtubules falls apart upon salt stress, okay? Remember that. Yeah. Now, if Veronia is important for this process, we would assume that a Veronia mutant would have the same kind of phenotype. And so to test that, we took a microtubule marker, introgressed it into the Veronia mutant, and you can see here when we treat seedlings with salt, the wild type copes relatively well with that, whereas in this double mutant, you can see that the microtubules are gone, and the same scenario for the Veronia mutant. So at least there's a link between how these mutants are responding to salt stress. Right. course, the ultimate evidence for the involvement of these phosphorylations by Feronia would be if Feronia can phosphorylate directly our protein. So we did actually a lot of assays to show this. Here's one such assay where you just express the active domain of our kinase together with our substrate, of course, the CC1, and add ATP, incubate that, and you do mass spec analysis to then check that, right? And from those mass spec analysis, we can show that there's at least 6 potential phosphorylation sites that are now added through this Feronia, right? The cool thing to me was that these phosphorylation sites are basically on top of where we previously showed that we have this microtubule binding domains. So this could actually be a neat mechanism for how this protein interacts with the microtubules. Of course, now we generated the obvious constructs. We mutated these 6 sites either to something called phosphonull, so basically amino acids that cannot be phosphorylated, or to amino acids that are mimicking phosphorylation, so to actually have it always on, right? And then we transform these constructs into this double mutant plants. Interestingly, none of those constructs rescued. So what you can see here is like when we transfer the seedlings to salt, wild type continues to grow, double mutant stalls. The wild type construct, of course, rescues, right? But all of these different phospho constructs do not. And just to, again, remind you of this mutant phenotype, when we now look at the microtubule arrays in these like phosphomutants, you can see that actually both in the null and the mimic, the microtubules are basically gone. So why is this? First, we were like, it doesn't make sense. It could be that, you know, the mimic is not really mimicking, but it could also be other reasons. And actually, I promised Brandon to show one Western blot, and here it is. Basically, what we did here was to pull down the CC1 with this YFP, and then we did basically a Western blot with this phosphoserine antibody. So basically, to see phosphorylation pattern of our CC1 during the salt stress, right? And what was really surprising to us is that actually before salt stress, we have a heavy phosphorylation of this protein. And when we begin to treat this, you can see after 2 hours, we reduce this phosphorylation substantially. And then that phosphorylation is coming back again. And of course, that is lost in the Feronia mutant because there you would not have the kinase anymore. So this actually kind of explains why the phosphomimic is probably not working, right? Because you need this dynamic phosphorylation pattern to be able for this protein to actually work properly. So just to explain it in a different way, typically under normal conditions, we will have a heavily phosphorylated CC1. And actually this makes a lot of sense because phosphorylation is inducing a negative charge in your protein. And the microtubules are net negatively charged. So they actually repel each other. And if you have a moving protein complex, of course, you don't want to have a tight interaction between your protein and the microtubules, right? It will basically putting a brake on the train on these rails. Yeah. So what is happening under salt is that you lose this phosphorylation, and this is increasing the affinity of our protein to microtubules. And we could show this in the paper as well. And therefore, you now begin to stitch back the microtubules together. And of course, now we're regaining the phosphorylation, as you can see up here as well, when you like continue to produce cellulose. Okay. So this is enough of nerding out on mechanisms, but I hope it's showing you that we can go from a completely unknown protein to probably the best characterized microtubule-associated protein in plant biology through these type of approaches. Right, so we can tell here that Feronia adjusts the phosphorylation of CC1 to control this microtubule behavior during salt stress. Last part of this, I'm going to introduce basically a new toolbox for analyzing protein-protein interactions in plant biology. maybe this can be of interest to you guys as well. But I would say that protein interactions was central to this whole project, right? Without that, we wouldn't really have these results. And maybe we can also take a step back, and most proteins we're working on, we don't know what the heck they're doing, right? I mean, of course, we can select proteins that are already known and continue to work on those. But most proteins, we have no clue. Even in Arabidopsis, about 1/3 of the proteins, we do not know what the function are of those. And so, of course, if you know what proteins are interacting, and one of those proteins is known, then you can infer the function for the interacting protein. They're likely to be involved in the same process. So it's something called guilt by association. You know, if you're associated with someone, you're guilty by just this association, right? There's many tools to analyze protein interactions, right? There's some that are really quite challenging, and you have to invest a lot of energy and money usually as well to do that. And then you have other types. You have, for example, standard techniques that are kind of screens and so forth, yeast-based assays, hybrid assays, right? You have co-IP, pull downs, etc., etc. But we all know that most of those actually comes with a lot of drawbacks. Many of those, I mean, in our hands at least, any split YFP assay, you can get anything you want to interact. And the same thing with yeast. I mean, I'm sorry, but the yeast-2 hybrid, it's great to confirm things, but I would be very hesitant to rely too much upon that. More recent advances have introduced something called proximity labeling. And so proximity labeling is basically outlined here. So you have your favorite protein of interest in yellow, right? And you fuse that to an enzyme through standard molecular biology techniques, right? And so this enzyme can then take a certain substrate here in black and put that substrate onto proteins that are close to your protein of interest. And then, of course, afterwards, everything that is now labeled with these black dots, you can enrich, you can mass spec that and find out what the heck it is. Most methods are actually using biotin bases for this. So this is basically a biotin ligase, if you want. And so this system, that comes with a lot of course, positive aspects. It's an optimized system. You can see all of these different enzymes over here have been developed. So TurboID is probably the one that's most frequently used currently, right? Because there's many users, right? So the optimization comes automatically with that. But there's some negative or basically drawbacks with this system as well. You typically, first of all, plants have biotin already, so you will have some background anyway. In many cases, that biotin level is not enough, so you have to add additional biotin. If you're working with seedlings, that's probably A-okay, because it's easy to add things. But if you're working with flowers or grain development, then it's relatively difficult to add stuff from the outside that gets into, for example, a grain, I would argue. Another drawback is that the substrate is released from the enzyme. So that means that these black dots are released from this enzyme before it's attaching to its substrate. So it will float away from the enzyme. So you would have some false positives in the system for sure. Another system that we are using is fully genetically encoded. Again, you have your protein of interest, called bait here, fused to an enzyme. In this case, it's called PathA. In this PathA, instead of using biotin, it will use a peptide. It's called PUP. And that PUP is put on lysine residues on proteins that is in close vicinity of your bait. So it's basically a ubiquitin-related system, but this comes from bacteria, and it's not present in eukaryotic species, so you will have no background. Right. Okay, of course, there's positive aspects and negative aspects. The negative aspect is there's less users. Currently, it's basically our group and Yanshin's group in plant biology. So there's certainly room for optimization, right? However, there's some really positive aspects. All of these components can be genetically encoded, right? So you can do this in your grains, for example. You can do it in whatever, right? Because you can just express both your enzyme and substrate. The second part that I think is positive is that the substrate, so this peptide, is maintained with the enzyme until it actually meets the protein, right? So basically these praise. So this protein here, so the enzyme has to physically interact with the prey to be able to put this peptide onto it, right? So there should be much less false positives in this case. So we have implemented this in a range of different systems now. So basically like in leaf infiltration in tobacco, Arabidopsis self-suspension, and also stable transgenics, both are actually Arabidopsis, rice, and poplar by now. And it works quite nice. So just as a sanity check, of course, I mean, before we're like basically selling this concept, we'll of course want to also make sure that we're getting some sanity behind it, right? So we actually like can identify known components of a protein complex. And so this is a protein complex that we discovered together with Daniel van Dan's group a long time ago by now, but it's called a T-plate complex after one of the proteins called T-plate, as you have here. But it's an 8-member protein complex that is the major driver of internalization of material in plant cells. So we use this as an initial example where we used T-plate as bait, and then we expected to see all of these guys, right, coming down or like being labeled by that. And maybe you can just remember EH1 and 2 and TW42 and 1, right? Because basically what we're now getting in this volcano plot here you have the enriched proteins after labeling. You can see that we're getting T-plate itself, making sense. We have the EH1, EH2, and we have the TW41 and 2, right? And you can see actually that the background here is quite clean. There's actually not much other proteins than the protein complex that we actually anticipated. Of course, the reviewer asked us then to compare this directly against the Turbo ID system because many people are using that. And this graph over here is basically more or less showing you how well enriched protein components towards compared to your bait are. And you can see that actually the Turbo ID is doing not that well because you have actually the lower the value here, the messier that you're getting basically in terms of labeling. But we are performing really well on track with basically APMS, which is basically a double enrichment pull down approach. So this was really satisfying to see. So of course we have done it as well for our CC protein. So coming back to that, there's a red thread through this talk. So we use these different constructs where we fuse the enzyme to our favorite protein, and we have this as a control. And of course we then have our substrate, in this case tagged with a flag that is easy to just pull down for identification of the proteins. And what we can see over here is that we get CC1 enriched, you have the different CSAs enriched, and you have this CSI1, which is cellulose synthase interacting 1 protein. So this makes a lot of sense in terms of this, right? Of course, there's also other proteins that you can see down here. But if we now look at the way that the CSAs are actually getting to the plasma membrane, it's getting through the ER to the Golgi and then trafficked up to the cell surface, then internalized and potentially recycled. So there would be a lot of proteins that are interacting with this protein complex. It's not as simple as the T-plate complex in this case. Actually, what was satisfying to see is that we actually got the T-plate in this. And you can see that we have T-plate, the WD42, 41, and so forth. And all of the blue ones here are part of this T-plate complex. And actually through, actually this is quite high-end super resolution microscopy with a spinning disk, we can actually show that this T-plate complex with this additional component surrounds this SA complex when it's internalized in a plant cell. So we can basically identify here components that are associated with SA complex when it's becoming internalized into plant cells. And of course, now to nerd out even further on this part, now there's about 10 to 12 people in my lab that works on this puppet assay. So we have a lot of data. And of course, what you now can begin to do is begin to assemble protein-protein interaction networks based upon proximity labeling, right? And so in this hairball again here, right, you would have now proteins and with each of the nodes, and all lines here now basically resemble like potential protein-protein interactions. And of course, the wider that edge is, the more experiments we have found this in. So we are building up now a proximity labeling based protein-protein interaction network. And currently we are implementing AlphaFold based analysis on top of this to actually like try to make sense of protein-protein interaction. I think this is something that is really well needed in plant biology in general to understand how proteins are interacting. Anyway, going to skip that. Yeah, the summary. Yeah, I guess abiotic stress impacts cellulose synthesis, in this case like salt. We also show that Feronia is an important regulator of cellulose synthase complexes during this process in salt stress. We have, actually this is something I skipped in this talk, so we can also skip that. But we have this new proximity labeling tool, Puppet, that I think will be very useful for protein-protein interaction analysis in general in plant biology. So with that, thanking my group, especially, where is it, Shuai, who was like developing the Puppet approach in plants in my lab. And there is the collaborators that we've had for the different projects that I showed in this presentation. So happy to take any questions, and I'm happy to also like end this conference. So thank you very much.
Yeah, thanks so much, Stefan, for the wonderful talk. Now, are there any questions?
Maybe I didn't have any questions because it's very vast presentation, but I want to learn. Especially we choose drought stress in sunflower. We make genomic analysis, but proteomic analysis is very huge job. So it is necessary all of us, especially my team. So we want to collaborate you.
Okay,
Okay.
we can definitely discuss that. So sounds good.
So, Alila.
Thank you. As far as this, I understand this labeling, it is really somehow similar to the Hi-C genomics, like you label what is nearby, but for Hi-C method, it's easy to sequence. You just go and you see, and how do you, I just don't know, how do you identify this labeled proteins?
So with the flag tagged substrate that we have, remember, you can use a flag antibody and just pull down everything that is labeled. Then you send it to mass spec, and there you identify the proteins.
So you know all the you know everything. Yeah, you know all the proteins.
I mean, actually, anything that is a sequenced genome, it's like relatively easy to even do proteomics on, right? You just have to map it on the proteome in that case.
Okay.
Many of the experiments are done in tobacco leaves, right? So there we're getting back the tobacco data, of course, right? Then we map that onto the Arabidopsis genome. So it's relatively straightforward, actually.
Okay, so it's like sequencing, because I never worked with it. And another question: so when you your one of the first slides, you showed us the transcriptomic data of co-expressed genes, and so I would expect this cellulose synthase would be somehow similar to housekeeping genes. It will be thousands of genes. But then you say, oh, we just took the CC1 because why? Well, how did you identify it among thousands of
That's a
genes?
very good question. Actually, Sue has also developed a lot of these like co-expression tools in the past, and we as well, of course. And housekeeping genes, yeah. I mean, primary metabolism is like a mess in terms of co-expression. I don't believe that you can really find anything useful from that. We were lucky enough, but of course we targeted perhaps 100 genes or so initially. But it's better to start out with that than 25,000, right? So at least you're like narrowing down the scope. I mean, it's the same thing with the protein interaction. Not all of those proteins will be directly relevant for what you want to do. So you still need to make a selection, and you base that selection upon what seems most likely in context of your process, right? So you will not be like, I'm getting one protein from this protein interaction. That's it, right? You will get like still 100 or so, yeah. But at least you can begin to sort through 100 instead of like the 20,000+, right? That you would have, or in barley or wheat, it will be many more, right? So, yeah.
Yeah, thanks again, Stefan. I have one question in regard of the cellulose synthase and the interaction with the microtubules at salt stress. Did you check out other stresses where, yes?
Yep. Those
same mechanism?
No, they're actually not responding to those. So we checked drought stress, for example. No response. I mean, we use PEG in this case, right? So I guess it's not really like drought stress, yeah. But we also checked heat stress and so forth as well, and there was actually no phenotype for those. So that that comment we got also from the reviewers, of course, they wanted to see whether this was a general kind of response to abiotic stress, but it does not seem like that. In the meantime, we have actually identified 2 other proteins from the same superfamily that also are associated with cellulose synthase complex. They seem to be involved in other abiotic stresses. So it might be that the whole family, it's actually LIA2 kind of domain on the outside. It's something that is like usually involved in stress responses and protection, right? So,
yes. Would be the next question about the LIA protein.
Right.
And maybe a naive question at the end from my side about the proximity labeling. You fuse it with biotin and then it's going on. And do you think that the fusion with biotin has an effect on if you take a possible steric hindering in account, because you're looking for
you mean in terms of functionality?
No, because you would like to check the proteins which are nearby your. But if you would then fuse, your protein of interest with biotin.
I can't hear. Can you?
So if you fuse it with biotin, that makes a difference.
Yeah, I mean, the biotin is actually just added on to proteins, right? So you have the fusion that you're making is with the biotin ligase. I mean, in our case, it's of course this peptide-related system, right? But overall, you fuse your protein of interest to an enzyme, and that enzyme is then taking a substrate to put onto other proteins. So of course, when you put that biotin or our peptide onto that protein, we don't know whether they're functional or not anymore, right? So that we don't know. But for us, it's not really anything we're really interested in, right? We're interested in just identify components.
Yeah. Thank you.
Yeah, thank you for presenting these exciting results. I try to anticipate the perspective of the plant. What is the benefit for the plant to stop cellulose synthase under salt and also to distinguish from other stresses? So to my naive interpretation, I would say the plant just tries to spare glucose for homeostasis, for more important things.
Yeah.
And but of course, it's somehow a smart way not to have one mechanism for all stresses, also to distinguish maybe for fine-tuning whatsoever. Would you agree to that?
Yes and no. I mean, I guess the why question is usually a very important, difficult question to address because we don't understand the plants that well, right? But I think also like the conditions that we're using in a lab is completely different than what you would have in the environment, right? So these proteins, we assign them certain function based upon observations that we do under ideal conditions, right? In the natural condition, it might be quite different in terms of how these proteins are actually acting. I think the mechanistic details of how it's acting, you know, in terms of binding microtubules and so forth, I think that is probably more robust in that sense.
Without interfering with salt tolerance all too much, because I assume it's a quantitative mechanism. It's not kind of digital thing.
I think to be honest, I think this mechanism will probably not be very fruitful to to implement in a crop plant. I don't think so. We try to overexpress the protein to see if it's becoming more efficient. Not really. So I don't think that this would be beneficial in a sense. Yeah, it's pure fundamental research to try to understand the biology. I think in terms of actually applied aspect, I don't think this would be that useful. So. Anything else?
Yes. Stefan Persson, thank you very much first of all for your very interesting report. I'm plant breeder working here at the Department of Plant Breeding and Seed Production. We usually select plants on their phenotype traits in the field, but your study is the same, maybe the same, but at the level of molecule. Could you watch there? I saw there were some seedlings under different doses of salt. They were different on their genes, different way as they develop under salt. Could you watch there? Was there any relation with the weight, with the English level, influence on the economic trades in the future the cars? Could you watch? Thank you.
Sure, thank you. As I said in response to Jochen's question as well, I think yes, we could probably do that, but I don't think that the mechanism I described here would be that relevant to salt trait in natural conditions. We're usually doing this on seedling or media plates, and I think it's very different than what you would have in the field. So we are trying to understand how biology works. That's our purpose with our studies. Then, of course, like some of the tools that we're generating, we hope that people can take advantage of in the applied field, right? But our goal is basically to understand how plant biology works. Yeah. So I think in terms of there's still like a bridge between the fundamental research and the applied research, and of course we are working to try to bridge that to some degree, right? But we still have a long way to go.
Firstly, thank you for your speech. And in your speech, you talked about interconnections of proteins, as long as I understand and understood. And you learned interconnections under salt stress, but you've also talked about that you also learned under other stress like heat tolerance. And I'm learning and I'm working in the sphere of plant breeding, which are which should be tolerant under high temperature. That the question is that is there any perspectives that learning protein interconnections or protein collaborations between each other? I mean, of heat tolerant under heat tolerant.
Sure.
So you get.
Yeah, sure. So we are doing some experiments as well where we take proteins that are associated with stresses, and then we fuse it to this enzyme that I talked about. And then you can treat your plants with or without stress and see how the protein interactions change. Yeah. So that will teach you a little bit about like how the important protein in that process is changing its function as well. So that's something that we are working on as well. We've been studying, for example, some kinases that are involved in sugar signaling, and then you can also get basically how are the interactions of that protein changes when it has sugar or no sugar. And actually, since that is a kinase, you can also look at the phosphoproteome. So basically the phosphorylation of the proteins that it interacts with through this system. And so that gives you a very nice way of getting potential signaling mechanisms for the protein. So hopefully that makes sense.
Then the first ever step would be learning phosphorylation, yes.
I mean, if you work on a protein that phosphorylate other proteins, then that would be important because that's usually how signals are progressing. Right?
Okay, thank you.
Sure.
Thank you.
I think, as I understand correctly from your question, you also want help from him if he could guide or like kind of collaborate to do something regarding your tolerance experiment or what?
O'zimni dissertatsiyam bo'yicha shu o'zimga taalluqli joyini so'rab oldimda. Kelajakda kerak emas, rahmat. Ketardik.
Okay, then I think, yeah. Thanks so much, Stefan, for the wonderful discussion and yeah, insightful results. And yeah, we can give big hand. Yeah, it's unbelievable. We come to the close of the workshop. Yeah, and now what to say, actually a lot to say. Yeah, to take this opportunity, I would like to thank each of you for your contribution to make this happen. And first of all the invited speakers and for sharing their excellent expertise by giving presentations. It's really, yeah, really exciting. And also for the participants and also I think your wonderful presentations and exciting discussions make this workshop really valuable and engaging really. Yeah, and also I want to thank to supporting institutions and Pushtaka and abroad. Please come here. Because without this support, it was difficult. Actually, the idea come up last year that we want to do like workshop, invite well-known scientists and kind of make it collaboration or make it a connection. Okay, then I think we can do it, but it turned out not easy. So I thought like I expected if we share on this idea, then everyone kind of excited and come to support us and okay, let's do this. But it was not as I expected, but anyway, we made it. So I'm really kind of thankful from everyone who helped directly, indirect way. Yeah, Shteke, nima aytmoqchimiz?
Ko'p narsa demoqchiman. Okay, first of all, I'd like to thank to lecturers who came from different parts of the world. And we know that the knowledge on biotechnology nowadays is very crucial to improve the crop productivity, tolerance, resistance. So this is important for better crop yield and a better life. Because we have in Central Asia, especially, big problems now, including the problem of the water deficiency, heat tolerance, and the other problems. So by the help of the modern biotechnology, we can solve very fastly and aimly we can fight against this kind of problems. So I will continue my some opinions in Uzbek. Nima demoqchiman? Avvalo, birinchi navbatda mana shu seminarga kelgan barcha haligi tinglovchilarga minnatdorchiligimizni bildirmoqchimiz, rahmat deymiz va alohida rahmatimiz, tashakkurimiz demak ma'ruzachilarimizni barchasi mana shu molekulyar genetika, molekulyar biologiya, biotexnologiyani dunyoda eng ilg'or markazlarini eng yetakchi mutaxassislari bu olimlar bilan biz hadeganda haligi aloqaga kirishib ular fikrini olish imkoniyatimiz yo'q bo'lmaydi ham. Bu juda ham katta imkoniyat. Avvalo mana shu seminarni tashkil qilishda asosan mablag'lar bilan yordam bergan hukumatimiz har xil fondlaridan yordam oldik. Rahmat deyishimiz kerak, tashakkur deyishimiz kerak. Sababiki bizga hozir biz o'zimiz bilimimizni kuchaytira olmaymiz. Albatta, O'zbekistonda ilmiy tadqiqot kuchayapti. Bu juda ham quvonarli holat. Zamonaviy bilimlar bizda ham mana ilg'or kuchayishni boshladi, lekin biz hozir dunyo darajasi bo'lishimiz uchun albatta mana bugungi kelgan ma'ruzachilar yordami kerak. Ular bilan biz birgalikda ishlasak, hamkorlikda ishlasak, yoshlarimizni jo'natsak u yerga, ular o'rganib kelsa, ular bizning kelajagimiz. Shu o'zimizdagi kelajakdagi hamma muammolarimizni hal qilishda shu yoshlarimizni ilmiy salohiyati bizga yordam beradi kelajakda. O'ylaymizki, bugungi o'xshagan seminarlar kelajakda ko'proq bo'ladi. Endi hozir albatta bizda haligi yozgi ta'til vaqti bo'lgani uchun biz vuzlar bilan haligi ko'proq yoshlarni jalb qila olmadik, lekin kelajakda endi hozir niyat qilyapmizki, bunaqa seminarlarni seriyalasak va bundan tashqari o'sha ko'proq talabalarni ham jalb qilsak, bu albatta o'zimizga, yurtimizga foyda. Hammalaringizga yana bir bora rahmat deymiz. Abrorga ham katta rahmat. Katta yordamchi bo'ldi. Bir-birimizga katta yordamchi bo'ldik.
Birgalikda. Assalomu alaykum, hurmatli mehmonlar. Sizlarni ko'rib turganimdan xursandman. I want to say thanks for our guests. I think our workshop is very informative and very successful. Barnose, yes, organize this workshop. It was very difficult, but we did this workshop. It is, I think, very necessary for us. All our presentation, it is very informative and very useful for us. Our republic is one of the important sector is agrar sector, because our population about 60, maybe more 60% our population work in agrar sector. Because aim of our research is developing this agrar sector. This biotechnology is, I think, it is future in this sector. I want to say thank you very much. Aim of this workshop is learn new technologies, take new informations, and I want in future collaborate with you. Not only me, all our institute wants collaborate with you. Thank you very much. Tashrif buyuruvchilarga kattakon rahmat. Kelib ishtirok etganlaring uchun. Xushtak ham ko'p narsa aytishdi. Bizning maqsadimiz faqat shu bitta institut emas, butun institutlar kelib, o'rganib, nima deydi, mana shu O'zbekistonni agrar sektoriga, mana ilm faniga rivojlanish, mana ko'rib turganlaringni, mana daklatchilarni juda yuqori darajada biz bular bilan hamkorlik qilib, faqatgina hamkorlik orqali shu darajaga yetishimiz mumkin. Agar biz o'zimiz harakat qiladigan bo'lsak, uzoq vaqt talab qiladi. Biz bu darajaga yetgunimizcha bu olimlar yana ham yuqori darajaga yetib ketadi. Eng yaxshi yo'li borib o'rganish, stajirovka, workshoplar tashkil qilish, hamkorliklar, loyihalar qilish juda muhim hisoblanadi. Shuning uchun shu vaqtlaringni ajratib, kelib, o'rganib, mana savol-javoblardan ko'rinib turib, qiziqib qatnashganlar uchun rahmat katta.
I wonder if anyone from the speakers or from the participant want to share their opinion and or impression about the workshop. If someone want to say something from the speakers.
Thank you, Barno and Abror and everybody here. It's been really wonderful to come to Uzbekistan and Tashkent and meet many of you. And I'm really excited to continue the communications and potentially collaborations. And I've met a few students here who are amazing. Arno is amazing. I feel very privileged to cross paths with Barno, and I'm looking forward to seeing her continue her success in her career, and she will. I have no doubt that she will become one of the leaders in plant science in the world, actually. And so I'd love to meet more Barnos in Uzbekistan, and this is my first time here, and my impression is really fantastic. And thank you so much for the opportunity and the privilege to come. It's been an honor. And I hope, like I said, that this is not the last time I will be engaging with you guys.
Thank you. Wonderful.
So for me, it's very easy. I fully agree what you have said.
Sure. I've also enjoyed the workshop, and I hope there will be more workshops in the future. And I hope that in future workshops, there will also be presentations from the local researchers here working in the two institutes and the university, so that we can communicate further and interact more with all the great research that's going on here. So thank you very much. I think this was a first step, and I'd like to see more activity in the future. Thank you.
Yeah, of course, I agree. There's not much more to add, I guess, what already has been said. But I also hope, in context of microscopy and cell biology, I hope this is also, maybe a good starting point to develop something advanced here as well. And I hope that will happen. I also have to say that I've been eating too much good food. It's making an impact, I think. So thanks. No apologies needed.
Yeah, I fully share what the other speakers have already said. My observation was that there are particular persons here in this room from Uzbekistan who are extremely interested. And when I have a maybe more broad perspective, it's obvious that your country needs biotechnology, plant biotechnology, as a breeding tool, because you had a lot of drought, you have too less water, and you have to cope with that. And this problem exists all across the world, but here, of course, it's a particular problem. And I see that we could contribute that via cooperation, via information flow, via communication, we can just make a better world and a better country.
Я, если вы позволите, говорю по-русски. Большое спасибо, Барну, за этот за замечательный воркшоп. Большое спасибо всем участникам. Спасибо, Курши, спасибо, Аброр. Спасибо всем, кто пришел. И я хочу, конечно, хочется сказать, мне нечего добавить к словам моих коллег, но на самом деле я бы сказала, что не только с нашей стороны мы можем внести какой-то вклад в дальнейшее развитие Узбекистана, но я абсолютно уверена, что Узбекистан, поскольку является действительно засушливым регионом, он несет в себе очень ценные генетические ресурсы для того, чтобы в дальнейшем обеспечить и помочь всему миру бороться с засухой. So I wanted to add to Johan's speech that Uzbekistan, as the same as Brazil, being hot region, definitely has lots of genotypes and genetic resources, which also could contribute for European development of this drought and heat resistance research. And I believe for future cooperation will be very productive. Thank you.
Я тоже скажу на русском. Я тоже поблагодарю Барна, Куршида, Аврора. Вы вообще сделали очень замечательную работу. В общем, пригласили столько крутых ученых со всего мира, и не на каждой конференции вообще можно услышать по целых 2 лекции, и таких полезных, и вообще современных. Это вот пик науки, пик биотехнологии, генетики. Это вообще очень ценно и очень здорово.
Bitta narsa aytib qo'ymoqchiman, uzr esimdan chiqib gapiraman deb. Demak, Barnoxonga ham bitta haligi chapak chalib qo'ysak nima uchun? Bu olimlarni biz o'zimiz olib kela olmaslik, ochig'i. Demak, Barnoxonning yordami katta bo'ldi mana shu sabab.
Hello, everyone, again. Let me speak firstly in my mother tongue. Assalomu alaykum yana bir marta hammaga. Demak, bugun mana afsuski workshopimiz mana 3 kunga bag'ishlangan workshopimiz tugab boryapti. Bu workshopda mana genom tahrirlashdan boshlab tortib u demak molekulyar o'simliklar molekulyar seleksiyasigacha strategiyasigacha bo'lgan nimani o'rgandik, juda ko'p narsalarni yangiliklar bilan tanishdik. Endi bu workshop mana hozir yuqorida biroz eslab o'tildi, anaqa oson bo'lmadi bu narsani qilish. Ular uchalovlarini anaqa asab buzilishlari, bu katta narsa, o'rtaga men ham tushib qoldim orada. Bular o'zimiz bilamiz u narsalarni, lekin nima bo'lganda ham men Barnoga, Xurshid To'raqulovga, Abror Qurbonovga o'z minnatdorchiligimni bildiraman bu workshopni tashkil qilganligi uchun. Bu workshopni biz 1 yil oldinlar taxminan demak rejalashtirgandik. Mana bo'ldi. Bu dunyodan ko'plab nimalarni, olimlarni, yetakchilarni olib kelish o'zi bo'lmaydi. Demak, thank you very much for our speakers from diverse countries, as well as from Denmark, from Germany, from United States, and from Russia, and from our brother country Kazakhstan. And I would like to thank in your mother language. So Gamsamnida, Dr. Sang Yanri, yes, and Mangtak, Brenda Wolf, and Stefan, and Danke schön, Dr. Johan Kumnen, and Michelle, and спасибо огромное, Sofia and Nastya, and Rahmet Kanatava, and very good, very excellent presentations, and very exciting discussions, not only here, I mean, not only presentations during presentations, and also in coffee breaks times, and lunch times, and I'm very exciting. Thank you very much.
Rahmat sizga. Kanat, you really sit with next to like a speaker. So I again you forgot and you didn't see it. So I'm 2 times. I'm really big. I'm really sorry, Kanat.
That's okay. So I'm not. Thank you very much. Yeah, let's say has been a fantastic 3 days. I'm happy to meet my previous colleagues and have a chat with them about the great science they're doing. I'm very happy, and just don't worry. I feel myself, as a local, so I don't get upset, and no worries, please. So what I can say is, whenever and wherever I meet any Uzbek brothers and sisters, I get quite amazed by their enthusiasm. So this is what we want to learn from you guys. Thank you very much for the exciting workshop.
Özür dilerim.
Öncelikle tüm Özbekistanlı kardeşlerimize, Kazak kardeşlerimize ve dışarıdan gelen misafirlerimize diyeyim. Hepsine çok teşekkür ederim. Buraya davet edildiğim için ve organizasyon komitesine, özellikle Ebrar Bey'e çok teşekkür ediyorum, Pamuk Araştırma Enstitüsü'ne. I want to be excited to be in here. They invited me as a speaker. It is a very big experience for me because I see many experienced scientists in here. So it is a very high level. I want this kind of workshop will be in the future in Turkey, the other countries, and I very excited the next step. Thank you, everybody.
What I can say at the end, I hope that the connection we made here will grow into fruitful partnership in the future. And thank you, all of you, for joining and making this workshop wonderful. Can we make again last photo? Yeah, group photo. Then after this we have lunch break. Yeah. The lunch break is the same place, right? Good.
That's good.